of nations to a very common man across the globe is being affected directly or indirectly with this coronavirus. This year, 2020, is universally recognized and remembered as the year of COVID. And with this great pandemic situation, the whole world has become paralyzed for months together. And everyone across the globe eagerly looking for a vaccine for this coronavirus. Scientists from various parts of the globe are working day and night to get the vaccine. And most of the developed and developing countries are striving hard and investing millions of dollars for developing the vaccine. Keeping in view of this pandemic situation, and as far as development of vaccine is concerned, as individuals, we have many doubts in our minds. In order to clarify these doubts, with reference to development of vaccine is concerned, this international webinar is being organized by involving the eminent resource persons from different parts of the globe who have been associated with this vaccine development. This webinar is an attempt to boost little confidence among ourselves to overcome with this burning issue of COVID. Hopefully, we expect that vaccine may come soon and we will lead a new normal life within a very short period of time. We are also ever grateful to our beloved Commissioner, Sir Sri Naveen Mittal Sir, who has been inspiring us for organizing a program like this. Without Sir support, guidance, and cooperation, it would not have been possible to take up this mega international webinar. We are very much delighted with the presence of our AGO sir, who has readily accepted our invitation to grace the program as a chief guest. I express my special thanks to Sri KSS Ratnaprasad sir, principal and chairperson of this international webinar, for his constant support and encouragement. I especially thanks Dr. Thiruvengalachari, Principal GDC Partala, for his guidance and encouragement. I also extend my thanks to Dr. Srimati J. Niraja Madam, Senior Academic Officer, CC, for support and accepted our invitation. We have got a tremendous response with almost 650 registrations from various parts of the country as well from other countries also. On this occasion, I express my sincere thanks to all the resource persons for their time and valid inputs. Thank you all.
Hello.
Unmute చేయండి మేడం.
Madam, unmute yourself, please.
Okay.
Again, I'll start. Okay. So thank you, sir. Thank you, sir. I'm very glad to invite our dynamic and friendly principal, Mr. K.S. Ratnaprasad, sir. Sir, you have hardly need any introduction. You have made all of us proud of your distinguished work in numerous capacities. I request you to express your valuable remarks on this occasion. Welcome, sir.
Thank you, ma'am, for inviting me onto the international webinar.
Yes.
From bottom of my heart, I thank the convener of the international convener, Dr. C.H. Srinivasu Garu and his dynamic team members. And my thanks to our dynamic Right Honorable Commissioner of College of Education, Sri Naveen Mittal Sir, IAS, and Dr. Balbhaskar Garu, AGO, CCA, Hyderabad, and ever helpful our Dr. Niraja ma'am, and the host of the participants. And I feel it a privilege to listen to the internationally acclaimed speakers on this very topic. This is very current, and everybody, as our convener said, everybody is on toes whenever and how fast we'll get the vaccine. Everybody looking after it. For it, for that, I thank all the participants for participating in this international webinar. Sorry again, feeling a little illness. Thank you all, and thank you, ma'am. Thank you very much. Please kindly take over.
Thank you very much, sir.
Thank you.
I'm profusely elated to take an opportunity to invite our chief guest of the day, Dr. Balabhaskar, AGO CC, Telangana. As we know, sir is famous for his administration skills, but many people like to remember him for his kind talk and smile with a grace. On behalf of our college, I thank him for providing his gracious presence to join us today to enhance our joy. So let us have the privilege of listening to the message of Dr. Balabhaskar Garu. Welcome, sir. We are eagerly awaiting your motivating words. Welcome, sir. Not there. Okay, okay. Sir will join us later. Dr. Balabhaskar, AGOCC, he will join us later. And now it's time for inviting the four pillars, means the four eminent and expert resource persons of this webinar.
Madam,
Sir. Okay, I'll
Madam,
continue.
sir was joined.
Neeraj Madam is there. Please call her to speak for a few minutes.
Okay, okay. Neerja ma'am is there. So I'll invite her. Oh, okay, okay. Sir is there. Balabhaskar sir is here. So welcome, sir.
Thank you, madam. Thank you. Thank you. Thank you.
Yes. So again, I'll invite him. Excuse me. I'm profusely elated to take an opportunity to invite our chief guest of the day, Dr. Balabhaskar Garu, AGO CC Telangana. As we know, sir is famous for his administration skills, but many people remember us for his kind talk and grace smile. And on behalf of our college, I thank him for providing the gracious presence to join us today to enhance our joy. So let us have the privilege of listening to the message of Dr. Balabhaskar Garu. Welcome, sir. We are eagerly awaiting your motivational words. Welcome, sir.
So good morning to all the members who are there on the meeting. It is a very great pleasure for me to be with you today on this opportunity, this occasion, like conducting an international webinar. I think this has been conducted by the Zoology Department of SR and BGNR College, Khammam. First of all, I must congratulate the organizers who are there behind this particular program. We are very happy. All our Telangana Government Degree College lecturers are doing well in the state by conducting so many webinars or seminars on this particular platform like Zoom. It is very happy for us to hear from you. I think this is the first time that our SR and BGNR College and other Telangana degree colleges, not only they are going into the state, but also they are going nationally, and as well, like these webinars, it is also going to the international way. Who are there, the ideas are what is our SR and BGNR college, all these things are going to be taken to now to the international platform. So I think this is a very good platform. because in the previous case, whenever we want to have any seminar or any webinar in the physical way, it is very hard for us to have such kind of things. Moreover, now the resource persons who are there, even they are coming from internationally to join with us to share their knowledge or to share their views, opinions over the topic, and that is the reason. So the only point is here, the webinars or seminars, I hope in the group, even students are also there. So whenever the participation of students is there, it will be very, very useful for the community, student community. I don't know the actual topic of the webinar. They just mentioned it is only an international webinar. So if at all, if you could give me what is the topic on which you are now conducting the webinar, I could able to understand the things. I can say that the thanks to the resource persons who are there from abroad to join this particular meeting. Now I request the organizers to just brief me on what topic this seminar is being organized. That is very important. I request
Okay.
Srinivasulu or any other organizer, please brief me what is the topic of the seminar today.
So I'll tell you the topic, sir. COVID-19, a pandemic of coronavirus, progress in therapeutic and vaccine development.
Okay. Okay. So it's a very, very good topic and very, this is the topic for the need of the hour. I think the seminar is going to be useful for the students as well for the participants who are there on the group. The COVID is, you all know very well that how the things are going now. The only one sector which is been affected with COVID is our education sector, because we have not been able to open even till today our education institutions because of this COVID pandemic. So I think the vaccine must come into the market. Once the vaccine comes only, I hope the normal life is going to be retained or restored shortly. So let us hope that we are going to have the vaccine in a short period of time. And the webinars like this also useful to know the status, what is going on in the research of the vaccine and other things. And these these webinars are also very much useful to the students to know how to take care of the COVID and other things, how to tackle the COVID. This is a very useful seminar, I hope. I once again thank the organizers who are there behind this to organize this seminar. I also thank the resource persons on behalf of our commissionerate for being joined with us today and to share their views and opinions over this. Of course, SR and BGNR College is an autonomous college. It's a big college, very reputed college. I also appeal to even the other departments of the SR and BGNR College to have such international webinars in coming days so that we can generate or we can create more and more confidence among our students, and we can also create more credibility about the college in future also. Because whatever the seats that we are giving at SNR and BGNR College, whatever the course it is, every student is opting the SR and BGNR College. That is the very special of the SR and BGNR College. Why the students are opting this college? Because they have the confidence on the college. They have the confidence on the staff who are working at the college. They have the confidence on the management or the principal who are there in the college. So that confidence only giving the more number of students to the college. So This is the situation that is there with the college? Only because whoever worked there in the earlier days, they have created such a great credibility to the college. So definitely now the present lecturers who are there, present professors who are there in the college, and also the responsibility is there on them to organize such webinars or seminars or such very innovative programs in the college, and so that they should take the name of the college nationally and internationally, and the college should also be in the one of the national ranking frame also. We are expecting that also shortly for this college. So a lot to do to the college. Again, there are so many things that are to be addressed in the college that we will definitely cooperate from the side of commissionerate to take the college again, to get this glory again into the student community. So definitely we will work on it. I appeal to all the professors or lecturers who are there working in the college also to take the responsibility of the college to work more to the college by organizing such webinars as well as seminars or any other innovative programs which are very useful to the student community and also to the community of the Khammam and other nearby this thing. I thank all the organizers for giving me this opportunity to be with you all today.
Okay. Thank you, sir. Thank you very much for your supporting words and
Kind words.
Thank you, sir.
okay. Thank you, sir. Thank you very much for your supporting words and for being with us. Thank you very much. Now it's time for inviting the four pillars of this webinar, the four eminent and expert resource persons. It's my honor to invite Dr. Mahendra B. Deval, obviously a PhD, and founding member and head of research and development in Expansion Technologies Inc., Cambridge, USA, to throw the light on the topic COVID-19, a pandemic of coronavirus, latest updates in vaccine and therapeutic development. I request Dr. Ravi Shankar, Assistant Professor of Zoology, to give an intro of the research person.
Good morning to one and all. On behalf of Department of Zoology, SR and BGNR Government Arts and Science College Autonomous, I, Dr. Ravi Shankar, have the pleasure on inviting our distinguished guest and today's invited speaker of the first session, Dr. Mahendra B. Deval, to this webinar. Dr. Deval is an alumnus of Giriraj Government Degree College, Nizamabad, and he completed his MSc in chemistry from Usmania University campus, Hyderabad. Then he briefly worked at IICT India at Hyderabad as junior research fellow with CSIR fellowship in a biotransformation laboratory. Then he moved to University of South Carolina, Columbia, USA for his PhD, where he has successfully completed his PhD in organic chemistry. He worked as a postdoctoral fellow in medicinal chemistry at Wayne State University. Then he moved to world's renowned institution, Massachusetts Institute of Technology, Cambridge, USA, where he worked as a research scientist in chemical biology. He is a founding member of Expansion Technologies, biotechnology company in Boston, USA. Presently, he is working as principal scientist, head of research and development at Expansion Technologies. Dr. Deval has scientific experience in multiple disciplines, including organic chemistry, medicinal chemistry, biochemistry, enzymology, molecular and cellular biology, chemical biology, genomics, transcriptomics, cancer biology, and expansion microscopy. He has 16 publications in high quality, high impact factors journals such as the Journal of American Chemical Society, PNAS, ACS, infectious diseases, and so on. He also has 4 US patents. Due to the time of constraint, I am not able to read out all his achievements like professional achievements, awards, presentations, and workshops, etc. Uh, as I have known him for the last 25 years, I would like to mention a couple of things about him other than his biodata. Usually, most of us are craving for private institutions to reach our goals. But we should note that Dr. Mahendra B. Deval had all his education here in public funded schools and colleges, then two in Telugu medium. One more interesting thing about him is he was introduced to English medium only from his post graduation. With brief, with this brief introduction of you, I hand over to proceeding to you, sir. Thank
Thank you. Thank you so much, Dr. Ravi Shankar, for a very nice introduction about me. Can everybody hear me? Just say somebody
Yeah.
yes.
Yes, sir.
Yes, sir. Yes, sir.
All right. Good morning, everyone. First of all, I would like to thank the Department of Zoology faculty members, Principal Sri KSS Ratnaprasad Garu, students of SR and BGNR Government College, Khammam. Also, I would like to thank convener of this international webinar, Dr. C.H. Srinivaslu, and coordinator, Dr. B. Ravi Shankar, who is a good friend of mine from OU campus days. Also, all the faculty members for inviting me to address today's international webinar. Thank you so much. And also, I would like to thank the chief guest, Sri Dr. Balabhaskar Garu, for gracing this event, and also all the attendees for gracing this event with your presence. Thank you. Thank you so much. Without a further delay, let me share my presentation and we'll get going. All right. I think everybody can see my presentation. Can somebody say yes or no?
Yes, sir.
Yes, sir. Yes, sir.
Thank you. Thank you.
Yes, sir.
All right. The title of my today's talk is COVID-19, a pandemic of coronavirus. Latest updates in vaccine and therapeutic developments. So, in the first half of the talk, I'm going to discuss about the evolution of coronavirus and its infection mechanism. Also, I'm going to talk about the components of the immune system and how they fight with virus infections. In the second half, I will focus mostly on vaccine and therapeutic developments, such as various different approaches used in developing the vaccines, and also latest updates and developments in therapeutics to treat the COVID-19. COVID-19, this 5-letter, 2-number word, has been shaking the whole world from past 10 months. What is this? COVID-19 is an infectious disease caused by the most recently discovered novel coronavirus. The coronavirus infections starts with most common symptoms, such as dry cough, sore throat, fever, diarrhea, headache, muscle or body aches, fatigue, nausea, vomiting, and tiredness. Also, as this virus infection keep growing in various different countries, some patients have reported 2 new symptoms such as losing smell and taste. Therefore, many countries included these 2 symptoms also as identification symptoms of coronavirus infection. As the virus infection gets into the severe stage, it may cause pneumonia, severe acute respiratory syndrome, SARS. That's why this virus is also called SARS coronavirus, causing the organ failures such as kidney and lungs, ultimately leading to the death. When I said in the beginning, a novel coronavirus, why this is called novel coronavirus? Is there any other coronaviruses existing already? Let's take a look into the history a little bit. The human coronaviruses were first described in 1960s for patients with the common cold. Here, I have listed 4 different serotypes of coronaviruses. 1 and 4th one. The first one and 4th one are described in 1960s, and the second and third one are discovered in early 2000s. All these 4 serotypes of coronaviruses are originated from either bats or rodents, but they are transmitted to the humans via an intermediate host such as camel or cow. These infections from these first generation human coronaviruses are very mild. That's why these viruses are not considered highly pathogenic. They only infect immunocompetent patients or people. However, in 2002, another coronavirus, SARS, severe acute respiratory syndrome coronavirus 1, broke out in China and spread to 5 continents, approximately 30 countries. And this coronavirus also originated from bats, but the intermediate host is either civet cat or raccoon dog, which transmits to the humans. And infections to the humans may cause mild to severe symptoms, also death. Approximately 8,000 people from these 30 countries were infected by SARS coronavirus 1, having 800 of them are declared dead. That's why this coronavirus is only limited to the 30 countries. That's why World Health Organization announced this as an epidemic, not the pandemic. Also, in 2012, another coronavirus, Middle East Respiratory Syndrome coronavirus, MERS coronavirus, broke out in Saudi Arabia, and this one also spread only 27 countries. So World Health Organization announced this also as an epidemic. A source for this coronavirus is also bats, but it is transmitted to the humans via an intermediate host, dromedary, which is also called Somali camel. And the infections from this coronavirus is also can lead to severe symptoms. The disease caused by this coronavirus is called MERS, or acute respiratory distress syndrome, ARDS. Approximately 2,000 people from these 27 countries were infected, having 700 of them are declared dead. As most of you already know that in 2019, SARS coronavirus 2, or novel coronavirus, broke out in Wuhan, China. It's very quickly, rapidly spread to the entire world, and World Health Organization declared it as a global pandemic by March 2020. Again, the source for this novel coronavirus, or SARS coronavirus 2, is also bats, but it is transmitted to the humans via intermediate host pangolin. And the infections from this SARS coronavirus 2 starts with mild, and it can lead to the severe and ultimately to the death as well. As of today, approximately 45 million people from all over the world are infected with this SARS coronavirus 2, having 1.2 million people are declared dead. United States, with 9 million people cases, is in the first place, and India is in the second place with 8 million cases detected with the infections of this novel coronavirus. So as of now, I explained 7 different serotypes of coronaviruses. Last 3 are seems to be much more dangerous than the first 4. So what are the differences and similarities between these 3 dangerous coronaviruses? Let's take a look at that. The most common similarity between SARS-1, SARS-2, and MERS coronaviruses is they are all originated from bats. The genomic sequence of all these 3 coronaviruses, 90 to 95% of the genome sequence of these all 3 coronaviruses matches similar to the bat coronavirus. And interestingly, the genome sequencing, 18% of the genome sequence of SARS coronavirus 2 matches with SARS coronavirus 1. That means they are 2 are brothers. They are from the same family. And 50% of the genome of SARS coronavirus 2 matches with MERS coronavirus. That means they are cousins. They are distant family. Another interesting feature between these 3 coronaviruses is SARS-1 and SARS-2 uses the same receptor, cellular receptor, to enter into the cell, which is ACE2, angiotensin converting enzyme 2. However, MERS coronavirus uses DPP4, dipeptidyl peptidase 4 receptor, to attach to it to enter into the human cell to infect the human body. So, however, among these 3 dangerous coronaviruses, SARS coronavirus is much more rapidly spreading compared to the MERS and SARS one. What could be the reasons? Let's take a look at that. The primary spread of these coronaviruses is person to person, either by coughing, sneezing, even speaking loudly. The droplets, the aerosols comes out of the mouth or nose, or they contains the virus particles and they cause the infections to other people. And the secondary spread also is another reason, which is contaminated objects and surfaces. If you look at this table, I have listed various different material or surfaces and the activity, the stability of the coronavirus 2 on various different surfaces at different temperatures. If you look at it at 4 degrees centigrade, that means in the fridge, the coronavirus active up to 2 weeks. That means it can still infect. But at 37 degrees centigrade, that means body temperature, but not inside the body. Outside the body, it is active up to 2 days. That means it's still infectable. But at 70 degrees centigrade, almost a boiling water, not quite boiling, but almost boiling water, it is deactivated within 5 minutes. And at room temperature, which is 22 degrees centigrade, the stability of this coronavirus varies, depends upon the material. If you look at that, the regular paper versus tissue paper, it is active up to 3 hours. And interestingly, currency note and cardboard, these two are also made of a paper, but the stability is different, which is up to 2 to 4 days, the virus is active, which is infectious. And also, if you look at the glass, plastic, stainless steel, these are also varies 4 to 7 days, the virus is active. That is also one of the reasons the rapid spread. And the third reason is long incubation period. Up to 2 weeks, the virus can be replicating inside, but the symptoms may not come out up to 2 weeks. This is another reason. The person who doesn't have any symptoms can also spread the virus to other people, infect the other people as well. That's another reason. Recently discovered, well, not recently, quite a few months ago, it was discovered that the mutations also causing this virus to be spread rapidly, which is particularly this single mutation, which is aspartic acid D at 614th position in this particular protein is mutated into a glycine. This is a spike protein of coronavirus, which has 2 subunits. One is S1 subunit and S2 subunit. This mutation is happening at the junction of these 2 subunits, which is much more closer view displayed here. This is the aspartic acid at 614th position in this protein, mutating into glycine. When this experiment conducted in the laboratory, so what they did was a plate of human cells were infected with a wild type coronavirus, which has aspartic acid at 614th position. And the second plate with the human cells infected with the mutated coronavirus, which has a glycine at 614th position. The human cells plate with the mutated virus much more rapidly infected compared to the wild type viruses. Also in the real-time data on the right side here, if you look at it, the mutation, in the January when the virus was announced in December, but the spread started in January mostly, but the mutation was not discovered until March. January and February, there's no mutation was discovered in the virus. This data came out of the virus genome sequencing from the patients who were infected in that particular period of time. In March, 26% of the patients were detected with mutated virus. In April, 65%, May, 70%. But now, more than 95% of the people who are getting infected have the mutated coronavirus. So, what is this coronavirus? So once Peter Medawar, he's an immunologist and Nobel laureate, defined in general virus as simply a piece of bad news wrapped up in a protein. Exactly. Like living cells, viruses also contain genome. In case of coronaviruses, it contains a long single-stranded positive sense RNA, which is securely wrapped by a nucleocapsid protein in a spiral shape. Also, this coronavirus has 3 other structural proteins such as spike protein, envelope protein, and membrane protein, which gives a very secured structure to this coronavirus. This spike protein also pokes outside the membrane, and because of these spikes, it looks like a crown. That's why this coronavirus is called this virus is called as a coronavirus. And one important thing is, unlike living cells or living bacteria. The viruses doesn't replicate themselves. They should not. They do not become 1 to 2, 2 to 4 like that, like the bacterial or mammalian cells. They require a human host cell to increase their population. And this coronavirus belongs to the zoonotic family of viruses, which means these viruses exist in the animals, but it can transmit animals to animals, animals to humans, and humans to human as well. When I said this genome, long single-stranded RNA is very securely wrapped up in the nucleocapsid protein. Let's take a look at much more closer look into the genome, how it is organized. The large genome, which is about 29,800 nucleotides, approximately 30 kilo base pairs of nucleotides encoding 29 proteins. This is the 2D map of the genome, which can be divided into 2 parts. In the first part, it has 2 open reading frames. Frame 1 and frame 2, which encodes 16 non-structural proteins of the virus. And second part has that encodes 4 structural proteins and 9 accessory proteins. Let's look at the functions of them. Among all the 16 non-structural proteins, only few of the functions of the few non-structural proteins known, such as NSP3 and NSP5. These 2 are the proteases. They help to cleave the long polypeptide chains of the virus to create its proteins. And this NSP12, which is RNA-dependent RNA polymerase, which is also called a replicase, very unique and very important enzyme for the coronaviruses or RNA-based viruses. What it does is it translates, it basically converts the positive sense RNA into a negative sense RNA so that the RNA can become more and more in the so that it can increase its population. It increases the replication basically. And NSP7 and 8 helps the replicase to function it very well. NSP1 has a unique property that I will explain you in upcoming slides. And the functions of the accessory proteins are not clearly known yet. And structural proteins, I already mentioned about the nucleocapsid protein. Spike protein plays a very, very important role in attaching to the angiotensin converting enzyme, the cellular receptor to cell entry, basically to cell enter into the human cell. Spike protein is very important. Envelope protein and membrane protein together work together, and they are the central organizers of assembly of the virus, and they gives a very stronger shape and robust shape to the virus. Now let's take a look at the cell entry mechanism. So when the virus binds to the ACE2 receptor on the cell using the spike protein, the cellular membrane engulfs the virus and the receptor together to form an endosome. Once the endosome is formed, then the spike protein goes through some structural changes and fuses into the membrane and releases the RNA, viral RNA into the cytoplasm of the cell. So if you look at this, this is a cartoon of spike protein attached to the virus membrane and which interacting with the ACE2 receptor on the cell membrane. Spike 2 protein, I already mentioned that it has 2 subunits, S1 subunit and S2 subunit. In this gigantic protein, which is a 1280 amino acid protein, this particular part, here is the 2D map. This particular small part, which is called receptor binding domain, plays very important role in binding to the ACE2 receptor. Once the RNA is released inside the cytoplasm, the first part, open reading frames, will be translated by the cellular enzymes into its non-structural proteins, which are the functional proteins such as replicase and all the proteases. When the replicase is already made inside the cell, it starts converting the positive sense RNA into the negative sense RNA. The negative sense RNA will be multiplied more and more by the cellular enzymes, as well as it will be created the mRNA of the other structural proteins and accessory proteins. Then this mRNA will be translated into the proteins, then transported into their appropriate places such as Golgi and endoplasmic reticulum, where they get refined, they get matured, and the virus assembly will start taking place at the membrane of the endoplasmic reticulum or a Golgi membrane. Once the full assembled virus is formed in the exosome, this membrane, here it forms an exosome, and then exosome reach to the cellular membrane. Then it will be released into the outside of the cell to infect the new cells. So as soon as the virus enters into the body, it attaches to the ACE2 receptor to infect the human cell or create the more cells. The first system in the human body response is immune system of the human body. Human immune system has two parts. One is innate immunity and second is adaptive immunity. Innate immunity comes by birth in each and every individual. This is like a frontline defense. It reacts to every toxin, every bacteria, every viral infection, anything, any wrong thing happening into the body, it will react non-specifically immediately within hours. And the innate immunity, the second one is adaptive immunity. So the adaptive immunity reacts slowly. It doesn't come by birth. It has to grow as we grow, as we age. Even though it reacts slowly, this is a backup defense. The very frontline defense is like innate immunity. It's like a police system, immediately attacks. The adaptive immunity is like a reserved army. It slowly activates, but it also specifically activates based on the antigens. But once it is activated, it lasts longer time, months and years, stays active and protects the body from various virus and bacteria and other type of infections. There is always a crosstalk between both the innate and adaptive immune system. And one more important thing is infants, babies, they're born with innate immune system, but they don't have the adaptive immune system. That's why they are more prone towards the infections. Also, as we grow, as we age, adaptive the cells of the adaptive immune system, their functions slow down. That's why older people, above 60 years, 55, 60 years age people, also more prone towards the virus or bacterial infections. So innate immune system contains many various different cell types. I'm not going to talk about all these cell types. I'm going to talk about very important, most important cell types now. First, what it does is, as soon as it detects the viruses or bacteria or any toxins, it releases some chemicals which are called cytokines and chemokines. They go to the infection site and releases the signals to the immune system cells, such as macrophages, nature killer cells, and antigen presenting cells. So as soon as they receive the signals from these chemicals, macrophages engulf the viruses. They eat the viruses and clears them out from the body. And nature killer cells, they activate the suicide mechanisms in the virus infected cells so that they can kill themselves. And as soon as the virus infected cells are dead, the macrophages clears them out from the body by engulfing them. While these two cells are doing their job, antigen presenting cells collect the antigen molecules from the viruses and virus infected cells, such as spike protein for the coronavirus, and takes them and runs towards the adaptive immune system to activate the adaptive immune system. Adaptive immune system contains two different cell types, B cells and T cells. T cells releases two different types of cells, T helper cells and cytotoxic T cells. Cytotoxic T cells, as soon as T cells receives the antigen molecules, they release these two types of cells. Once the cytotoxic T cells has the same function as the nature killer cells, they basically kills the cells, activates the suicidal mechanisms. But T helper cells generate the memory cells. What the memory cells does is they remembers the antigen molecules on that particular virus. This is a coronavirus, a dengue virus, such as all these different virus antigen molecules they remember, and they even remembers the fighting mechanism of that particular virus and remains as a reserved army in the body. Whenever that virus comes back into the body, they will fight with it and clears them out. And also B cells, what they do is they create the plasma cells, which generates various different types of antibodies, including the neutralizing antibodies. What neutralizing antibodies does is they basically arrest the cell. They bind with the spike for coronavirus. They bind to the spike protein on all over the places so that it will not go and bind to the ACE2 receptor to infect the human cell. So as soon as the neutralizing antibodies bound to the virus, the nature killer cells or cytotoxic T cells come and then eats them or kills these viruses, and macrophages engulf them, clears them out from the body. However, interestingly, for somehow the coronavirus is escaping this human immune system's attack and increasing its population in the human body. How there are various different hypotheses are described in the literature that I'm going to explain to you in a minute. But very first thing what it does is it sends the wrong signals to the immune system, so that immune system releases various different cytokines, many cytokines. The cytokines are two different, anti-inflammatory and pro-inflammatory. It generates lots of pro-inflammatory, which causes damage to the tissue or cells and organs. That is called cytokine storm. That's how the organ damage to, as soon as the cytokine storms into the body of the particular cells, and the organ damage or cell damage or tissue damage leads to the organ failure and death. That's how lots of people are dying because of these coronavirus infections. So there are hypotheses that coronavirus escapes from this immune system's attack. One hypothesis is accessory proteins may be helping the coronavirus to escape from the immune attack. How? It's not quite clear, but there's a hypothesis. The second hypothesis is conformational flexibility of the spike protein. Spike protein exists in two different conformations. One is open conformation. Second one is a closed conformation. In open conformation, the RBD, this is in the green color, and rest of the S1, S2 subunits are in here. The RBD is in a standing position, and which is the active position, which is ready to attack on the S2 receptor, enter into the human cell. This is the more active conformation. And this is the sleeping conformation, where the RBD is in closed form, which is a sleeping position, which is not able to attach to the ACE2 receptor, cannot enter into the human cell. So when the immune system is trying to find the virus, it is probably forming this closed conformation and escaping from the immune system. This is another hypothesis. And the third hypothesis here is that the sugar coating on the spike protein is heavily glycosylated. Glycosylation is a concept where the sugar molecules, the long chain or complex sugar molecules are attached to the proteins to give a nice, strong, robust structure and functional properties and solubility, stability to the protein. So when it is heavily glycosylated, then the RBD or the spike protein is not identified by the immune system proteins, and it can that's how virus is escaping from the immune attack. Recently, a month ago, a new new discovery was made, which is NSP1. I mentioned in my previous slide, but NSP1, non-structural protein 1, is helping the coronavirus to escape from the immune attack. This is how it is happening. It is inhibiting the translation of mRNA of the immune system's proteins, immune suppressor proteins, basically. So this is a ribosome. This is a tunnel where mRNA translation takes place. mRNA will be translated into the protein and goes back into these functions, right? It binds exactly at the edge of the tunnel, where this is the mRNA comes here, and at the 3' end, it goes outside, and then as a protein, it blocks it right here and stops the translation of those proteins, so that the immune suppression proteins are not generated, so that the virus can grow its population more and more. So when this virus takes over the body like this, that's how we need support from outside. When body is not able to protect, not able to fight, not able to have enough energy to fight with this infection, then we need the support from outside, such as vaccine or therapeutic development. Now I'm going to talk about the vaccine and therapeutic developments. So the vaccines, first option is vaccines. What vaccines does is they don't cure the infections, virus or bacterial infections. They just prevent them, or they protect from the infection, or either they protect from the infections or they prevent infections. When vaccine cannot cure, this is once the virus enters and this is infection takes over the body, vaccine cannot do anything, and that's place where we need the treatment options such as antiviral therapies and antiviral agents. So first, I'll start talking about vaccines, their developments, and then this clinical stages and so on, then various different approaches. Then I'll come into the treatment options. Before we go into all these steps, first, let's understand how does a vaccine work. Vaccine could be a virus, or a part of a virus, or a mimic of a virus that mimics this particular infection. As soon as you inject the vaccine inside the body, it releases the antigen molecules, which are identified by the antigen presenting cells of immune system, adaptive immune system. Basically, it strengthens the adaptive immune system. Antigen presenting molecules, antigen presenting cells take these antigen molecules and runs towards the adaptive immune system and activates the T cells and B cells, which releases the neutralizing antibodies and memory cells that will be reserved as a reserve army whenever the virus infection comes into the body to fight successfully with the virus infections, these memory cells and neutralizing antibodies generated by the vaccine may stay there from months to years to protect and fight with the viral infections when they come into the body. So any virus, any vaccine or drug discovery process takes, there are two different stages, preclinical stage and a clinical stage to develop a vaccine or a drug molecule. In the preclinical stage, the molecule is developed in the laboratory and tested, uh, in the human cells, and then move into the animals, tested for the safety in the animals. After that, once it passes the animal stage, then probably if there is a chance, one or two healthy individuals can be tested the safety of that vaccine or a drug molecule. Then it moves into the clinical stage. In the clinical stage, there are 4 different phases: phase 1, phase 2, phase 3, and phase 4. In the phase 1 stage, less than 100 people, healthy individuals, will be tested for safety and dosage. Dosage, for example, how much should we give? 25 micrograms, 100 micrograms, 50, how much vaccine we should administer into the body, so into the person. 100, less than 100 people will be tested at the phase 1. Approximately 70% of the candidates pass the phase 1 clinical stage and move to the phase 2 clinical stage. In the phase 2 clinical stage, approximately more than 100 people will be tested for the efficacy and side effects. I hear some kind of there is some noise I'm hearing. Okay, that's fine. So in the phase 2, the more than 100 people will be tested, approximately 300 people will be tested for the efficiency and side effects of the potential vaccine candidate or the drug molecule. And these 2 stages will be conducted, these 2 phases will be conducted between age groups between 18 to 55, and that to healthy adult individuals. Approximately 33% of the potential candidates move into the phase 3, where thousands and thousands of people will be tested for the safety, efficiency, and adverse reactions of that potential vaccine or drug candidate. That too in not only the 18 to 55 age healthy adults, but also more than 55 years old, older adults will also be tested, including people with pre-existing conditions such as diabetic patients or dialysis patients, heart patients, or diseased condition patients will be tested. And then at this stage, regulatory review will happen. Once the regulatory review is passed and approved, the drug or potential candidate will be released into the market as an investigational medicine, where the phase 4 will start into the real market, and it will be monitored for long-term side effects. 25% of the potential candidates moves into stage 4, and less than 10% of these the drug molecule or vaccine candidates will come to the final complete approval, so that they can administer it into any individual, including infants or babies, children or older adults or young adults, anybody, diseased people, anybody. So less than 10%. This entire process takes place approximately 12 to 15 years. The preclinical stage itself takes 5 to 6 years, and clinical stage approximately 7 to 9 years. However, because of this current pandemic situation, regulatory committees, regulatory authorities are decided to give some allowances, such as they can conduct all these phase studies simultaneously to speed up the vaccine or drug discovery process. So in the vaccine development, there are various different methods are used. There are conventional approaches, such as using viruses as vaccines, or a protein from a virus using as a potential vaccine. And there are next generation platforms are available from past 3-4 decades. In olden days, 60, 70 years ago, these are the only 2 approaches were there. But recently, next generation platforms are also developed, which uses viral vectors, which are engineered viruses, and also nucleic acids or genomes, genetic material of the viruses also can be utilized as a potential vaccine candidate. To develop a vaccine candidate, antigen molecules are very important. For coronaviruses, structural proteins play a very important role as an antigen molecule. Most importantly, spike protein is being very widely used to develop the vaccine, potential vaccine candidate for the coronavirus infections. As of today, according to World Health Organization, there are 201 potential vaccine candidates are in development from various different labs and companies, organizations throughout the world. Among these 201, 45 candidates are already in human clinical trials at various different stages that I will explain you in my upcoming slides. First approach, the conventional approach, is using the viruses as a vaccine. There are two different methods. Using the live viruses, but they are weakened. They are attenuated virus. For example, coronavirus can be utilized, but it has to be weakened. There are different methods used to weaken the virus or attenuate the viruses. They can be cultured in the chicken egg or other eggs, or they can be passaged into human cells or animals so that they can become weak. Because viruses, with the passage in generation, they mutate constantly, regularly. The mutations can be bad or good in the sense of for the virus. When bad mutation happens to the virus, it becomes weak and weak. So that virus can be utilized. Weak virus can be utilized as a vaccine candidates. Here are some examples: smallpox virus, measles, mumps, rubella virus, chickenpox virus, influenza viruses are successfully developed and being used currently in the world that using this live attenuated virus vaccine method. For coronaviruses, Indian Immunological Limited, Serum Institute of India, and Codagenix, these companies are also trying to develop a potential vaccine using this particular method for coronavirus infection. They are at preclinical or phase 1 clinical trials at the moment. The second method is using inactivated viruses. Viruses can be inactivated by either heating or using a chemicals such as formaldehydes. Once the virus is basically killing the virus, it kills, but the virus parts are intact. That can be utilized as a potential vaccine candidate when it is these inactivated virus is injected into the body, or attenuated virus is injected into the body. It will strengthen activates the immune system and produces or induces the adaptive immune system and strengthens the adaptive immune system. Poliovirus, hepatitis A virus, rabies viruses are successfully developed using inactivated virus method. Presently, Bharat Biotech from India, Sinovac and Sinopharm companies from China are using this method to develop a vaccine for COVID-19 or coronavirus too. They are conducting phase 1, 2, 3 clinical trials simultaneously in various different stages at the moment. Second method, conventional method, is using a virus protein. Excuse me. For example, coronavirus, spike protein is a much more widely used antigen. A spike protein can be utilized as a potential vaccine. It can be formulated in a lipid nanoparticles or another formulation, and it can be injected into the human body, or only receptor binding domain. RBD can also be utilized, or only S1 subunit can be also utilized to create a vaccine to induce the immune response. Seasonal influenza vaccine was successfully is the best example, successfully developed using this method, and presently is being used widely all over the world. Novavax, a company located in the United States, is using this approach to develop a vaccine, which is NVX-CoV V2373. They are using a lipid nanoparticle formulation in addition to matrixin adjuvant, their proprietary adjuvant, which boosts the immune system. It's an additional help. They are developing the vaccine. Currently, all 3 phase clinical trials are underway in various different countries. They also released interim data which showed very good results, induced the immune response in all the participants without any side effects, that too in healthy individuals at the moment. Still, the phase 3 trials are ongoing. Next generation platforms, the other approach is using the viral vectors to develop the vaccines. Excuse me. In this approach also, there are 2 different methods. Using the replicating viral vectors, such as weakened live attenuated viruses, the weakened measles virus. That means their genome will be engineered, such as it can express the spike protein to generate a vaccine for the coronavirus, but it will replicate as a normal virus. But all the other genes of that particular virus will be silenced or deleted. And the second approach is, excuse me, non-replicating viral vector. This virus, again, the genome, entire genome will be engineered. The spike protein gene will be inserted in there, and rest of the virus gene will be silenced or deleted. And especially the replicating replicase enzymes also will be deleted so that this virus cannot be replicated. This is another method. When these two vectors injected into the body, they will also induce the immune response. CanSino Biologics, one of the first company from China, started working on this next generation platform using adenoviruses. This is the most common viral vectors are used widely from past 3 decades to generate the vaccines or other drug molecules. The adenoviruses cause only common cold in the human. They are harmless, not that harmful. That's why they are very potential vectors to develop a vaccine. Within 3 months, December, pandemic started. By February, they completed the preclinical studies, which is very fast. Usually, it takes about 5 to 7 years. And they moved into phase 1 March. Currently, all 3 phase 1, 2, 3 clinical trials are underway. They have released the interim data of the phase 1 and phase 2 clinical trials, which looks very promising. And approximately 50 billion viral particles are 1 dose. They injected into the humans to generate sufficient immune response. The other companies, such as University AstraZeneca, together with the University of Oxford, they're also using an adenoviral vector, but this is not the CanSino Biologics using a human adenovirus. But AstraZeneca and Oxford, they are using chimpanzee adenovirus. They're to develop a vaccine for COVID-19. They also released right now phase 1, 2, 3 clinical trials are ongoing already. Another company, Genesen, a pharmaceutical company of Johnson & Johnson, also using adenovirus, the human adenovirus, which is a 26th serotype. CanSino Biologics used a 85th serotype of that adenovirus to develop the vaccine. Both of the companies are conducting successfully completed preclinical studies, conducting right now the phase 1, 2, 3 clinical trials. AstraZeneca is way ahead of in the race compared to any other, all the other viral vector vaccine companies. They also released interim data which produced a rapid immune response in 90% of participants without any side effects. Their goal is to produce 1 to 2 billion doses globally by next year. However, one caveat we still have to keep in mind that until today, no vaccine was successfully developed using adenoviral vectors. This is another important factor we have to keep in mind. Though, let's keep the fingers crossed that one of these companies will be successful in soon. The next generation platform, other next generation platform is nucleic acid based vaccines. A viral DNA, if you look at here, spike protein, again, antigen molecule, S1 subunit or S2 subunit or receptor binding domain. The DNA encoding either receptor binding domain or entire spike protein can be injected straight into the human muscles to induce the immune response as a potential vaccine candidate. Or the RNA encoding either spike protein or RBD formulated into some formulation and injected into the body to induce the immune response. RNA-based vaccines are developed by, I listed only 2 important companies here. Moderna Therapeutics, they are using mRNA 1273 together with National Institute of Health, American government agency. And also Pfizer together with BioNTech and Fosun Pharma also using an mRNA-based vaccine. What they're doing is they're encapsulating the mRNA encoding the spike protein into a lipid nanoparticle and administer them into the muscles to induce the immune response. The Moderna already released phase 1 initial and final results, which showed very promising results. This is another company that is very ahead in the vaccine development. Hopefully it will soon. And another thing is this company also tested in older patients above 55 age group, which showed very promising results. BioNTech started with 4 different mRNA candidates. Unmodified mRNA, which encoding the entire spike protein, modified mRNA encoding the only receptor binding domain, modified mRNA encoding complete spike protein, and self-amplifying mRNA encoding complete spike protein. Among all these 4, BNT162b2 spike protein encoding mRNA, modified mRNA, won the race, and now it has been going through all the clinical phases 1, 2, 3 trials. And this month, they actually submitted a rolling review to European Medical Agency for an approval process. Basically, the approval process begins with this rolling review for either at least an emergency use authorization. But it takes time to get the results, the committee results. And DNA-based vaccines. The DNA-based vaccines, Inovio Pharmaceuticals is developing using a spike protein DNA. They have a proprietary device, a Selectra, which transmits the virus into the skin within seconds using the electroporation method. So they have a proprietary using this instrument. They formulated the DNA. They're injecting the DNA straight into the skin to induce the immune response against the COVID-19. Right now, they are conducting the phase 1-2, successfully completed the preclinical study, and phase 1-2 are simultaneously ongoing. And another interesting and important point here is this is the only company has a potential vaccine candidate for MERS coronavirus in phase 2 clinical trials. However, again, this is a next generation platform. No vaccine has been produced using the nucleic acids till date since the platforms are developed. This is one of the caveats we have to keep in mind. Hopefully, one of these companies will get a success. So lots of people are asking what stage, which company is there among 45 potential vaccine candidates are in the clinical trials. So I have summarized all of them in this table. So I'm going to explain you cleanly. Sinovac, this is a Chinese company. They are using inactivated virus to develop the vaccine, which is CoronaVac. And it requires at least 2 doses. First dose is given in the day 1, and then after 3 weeks, second dose will be given to induce a sufficient immune response to clear the vaccine. And they're conducting clinical trials in multiple different countries, which is also important. The clinical trials has to be conducted in various different regions, various different countries to make sure the vaccine is efficient, safe. Bharat Biotech, one of the Indian company, they're also using inactivated coronavirus to create a vaccine. And they are doing phase 1 and 2 clinical trials only in India. They also require 2 doses. Except the CanSino Biologics, Oxford and AstraZeneca, rest all the vaccine candidates require 2 doses. Only these AstraZeneca and CanSino Bio requires only 1 dose to induce sufficient immune response to clear the coronavirus from the body. CanSino Biologics, I already mentioned in here, they are also conducting multi-country clinical trials, all 3 phase clinical trials. They are using non-replicating adenovirus vectors. AstraZeneca also conducting various different countries. This is way ahead, I mentioned, compared to the various different candidates. Europe, USA, India, Japan, Russia, Africa, and Indonesia. All these countries, they're conducting phase 1, 2, 3 clinical trials. Basically, they're conducting 1, 2, 3 trials in Europe, but phase 3 in all various different countries. It also requires only 1 dose. Johnson & Johnson, we have multiple different countries. It also requires 2 doses. First dose on the day 1, and then after almost 2 months, we give a second dose to induce a sufficient immune response. Gamaleya Research Institute from Russia, they also developed adenovirus vector-based vaccine. They're using 2 different adenovirus vectors, AD5, which is used by CanSino Biologics, AD26, which is used by Johnson & Johnson. They are using 2 doses, 1 dose this one, and second dose is AD26 to induce a sufficient immune response. But they are conducting clinical trials only 2 countries. Inovia and Zydus Cadila in India also developing a DNA-based vaccine, which requires 3 doses to induce sufficient immune response, and they are only conducting clinical trials in India. Moderna, I already mentioned. For Pfizer BioNTech, I already mentioned, which also required 2 doses. They're also conducting multiple countries, except Moderna. Pfizer is conducting clinical trials in multiple countries. Novavax protein-based vaccine also conducting clinical trials in multiple countries with the 2 doses required. So one important thing we need to consider here is, even though vaccine is available, single dose is fine. If it is required for 2 doses, and then the production need to be doubled. That means the companies need to work much more efficiently to create a vaccine to produce more and more to sufficient that all the population, world population, can be immunized using the vaccine. And among all these front runner candidates I have listed here in this slide, three candidates in my mind, which is Oxford, AstraZeneca, Moderna, and BioNTech, are way ahead of any other companies. So Moderna and BioNTech, I already mentioned that they have started the rolling review process of the for the regulatory review, and Moderna also is going to start anytime from November or December. So they may be available as an emergency use authorization early next year or mid next year. Hopefully, let's hope for the best, that one of the vaccine candidates will be available for the use. Now I'm going to quickly wrap it up. My apologies if I'm taking a longer time. In 2-3 minutes, I will wrap up the therapeutic developments. In the therapeutic developments, there are two different methods used: repurposing the existing therapeutics, which is screening all existing broad spectrum of antivirals against the COVID virus. That's one method. Second is other molecule existing drug database, blood pressure drugs, cardio drugs, or renal drugs. All drugs can be screened against the COVID-19. The second approach is developing from the scratch, using the genomic information, new targets from the base, which is going to take years and years, much time consuming. But repurposing is much quicker and faster. As of today, there are approximately 427 potential candidates are under development, and 340 of them are in clinical trials. These are the drug molecules that are came out of the repurposed when the antiviral agents, broad spectrum of antiviral agents, are repurposed against COVID-19. These molecules came out. So hydroxychloroquine, leponavir, ritonavir, these two drug molecules are protease inhibitors. They are used for HIV. All these three molecules later on, initially scientists or doctors thought that they are using and they're helping to recover the patients, but later on in the clinical trials they found that they are ineffective, therefore they are discontinued. These molecules are discontinued. Drug molecules are discontinued from the treatment. And these three molecules are also hepatitis C, influenza, approved for the influenza treatments, and hepatitis C treatments also showed some promising results. They're presently going through clinical trials. And remdesivir and favipiravir. Remdesivir is recently approved by FDA. This is a repurposed drug. Originally, it was developed to treat Ebola infections, and it was only in the clinical trials. And when it is repurposed against COVID-19, it showed a promising results, and the clinical trials successfully completed and approved by FDA. Favipiravir, right now it's been approved for an emergency use authorization in India by Drug Controller General of India, and this also influenza, hepatitis C, and Ebola virus infections can be treated by favipiravir. So remdesivir is being marketed by Gilead as a Veklury, approximately 100 milligram injection. Cipla and Hetero are marketing in Cipremi with the names Cipremi and Coifir in India. It started its clinical trials. It's repurposed. This is the only drug as of now today approved against COVID-19, the first drug approved against COVID-19 treatments by FDA in the US. The clinical trials started in March. By October, it was approved, approximately 6 months, which is a very fast pace. Here is the structure of the drug molecule. When it is enters into the body, this is a prodrug, and this is the original drug, which is a mimic of nucleotide adenosine in the body. And this is the crystal structure where it binds to it basically blocks the replicase activity of the coronavirus, where replicase converts the positive sense RNA into a negative sense RNA. It binds to the RNA and blocks binds to the enzyme and blocks its activity. Favipiravir, again, I mentioned already that it has been approved as an emergency use. Here is the prodrug, is the original drug. Presently, phase 2-3 clinical trials are underway. Hopefully, anytime it can be approved. And Glenmark is marketing in India for emergency use authorization. Another novel molecule, this also came out of the drug repurposing, which is EIDD 2801, molnupiravir, was developed to treat Ebola virus infections by MRI University together with Ridgeback Bio and Merck Pharmaceuticals. This molecule also blocks the replicase activity. It is also a nucleotide mimic, which mimics the uridine. And one interesting thing is this can be taken as an oral pill. Currently, phase 1, 2, 3 trials are ongoing. Anytime the data may come out, and which may be, hopefully, it can be approved to treat COVID-19 infections. The other approach is hyperimmune therapies, which my lab is working on. So the hyperimmune therapies, the antibodies will be extracted from the convalescent patients, the convalescent plasma, and modified. So if you look at this antibody, here, this yellow one, antibody binding to the RBD, which supposed to block the interaction between the ACE2 and RBD so that the virus cannot enter into the human cell. But this antibody binding in a different way. What we do is we take these antibodies and use some machine learning algorithms to modify them and synthesize them in the lab and test them in the pseudovirus, then real virus, and to make them much more potent antibodies to treat the COVID-19 infection. So far, I have discussed about the evolution of coronavirus towards the COVID-19 pandemics. I've discussed about the human surveillance system, immune system, its components, how it fights with the viral infections, how virus takes over the immune system, then the development of vaccine and therapeutics. However, so far, all these three coronavirus, dangerous coronaviruses are broke out: SARS-1, SARS-2, and MERS. We don't have any vaccine, neither either SARS coronavirus 2 or SARS coronavirus 1 or MERS. None of these viruses does have a vaccine. So how can we survive until then? So most important thing is we have to live like we're living with the virus, and we have to avoid exposure to it. So to do that, regularly, must and should we should wear the mask, much more efficient. Frequently washing hands, sanitizing hands is important. Practicing the social distancing is very, very important. Most important thing is avoiding the social gathering. These are much more effective than any therapeutics to prevent or cure the COVID-19 infections, or not to infect more people. Also, an additional thing is boosting the immune system. Eating healthy or nutritional diet, sleeping, getting enough sleep, active lifestyle, getting enough exercise will activate your immune system, strengthens your adaptive immune system, so that adaptive immune system will successfully fight with the viral infections to clear them out from the body. So in this journey, my journey started from OU campus, Department of Chemistry, where I started learning chemistry. Then I briefly worked at CSIR IICT, where I started my first reaction. I'm exposed to the research environment, research lab. I always remember all the wonderful colleagues and friends over there. Then I moved to Department of Chemistry of University of South Carolina, where I completed my PhD in organic chemistry. Then where I learned organic synthesis, making the molecules and testing them on materials, various different materials. Then moved to the Wayne State University, where I worked on the medicinal chemistry platform, making the molecules, then test them on the bacteria. I learned lots of molecular biology and biochemistry skills, working with the bacteria. I learned here, then moved to the MIT in chemistry department, where I started working with the mammalian cells, viruses, HIV, all various different types of viruses, using my skills learned various different places to prevent the cancers and virus infections, research that I have done over there. Presently, I work at Expansion Technologies, where we are developing new early stage diagnostic methods to detect the cancer in early stage, various different cancers such as prostate, breast, lung, kidney, and also I'm working with human tissues down here. Having said that, all the people here, my teachers, my colleagues, my friends, my students, everybody helped me to grow as a scientist. I have to thank each and everyone, but not the least, my family and my friends, who also helped me a lot. Without their unconditional support and encouragement, I don't think I would have been where I am, what I am today. Having said that, thank you so much for your patience. I appreciate it. My apologies, I took 10 minutes over. Thanks again, once again, and I'm happy to answer any questions if you have.
So thank you, sir. I think our participants posted their doubts in the chat box. I think you can-
Please go ahead. I'm happy to answer.
Yes.
Can somebody read them to me?
Hello, good morning, sir.
Sir,
Or somebody can unmute and answer, ask a question on the Zoom platform also. Whoever is on the Zoom can unmute themselves and ask questions.
yes, yes.
Sir, good morning, sir.
Good morning. Can you
Sir,
please state your
I
name?
have one doubt. I am M. Venkat, working SR and BGNR Degree College, Khammam.
Mhm.
I have one small doubt, sir.
Please go ahead.
Can single virus can destroy our human immune system?
Absolutely right. A single virus is sufficient to make more and more virus. Once it enters into the body, it is not
Yeah.
single anymore.
Yeah.
It will replicate it. I mean, our body helps the virus to make more and more. Because basically what virus does is it enters into the body, it hijacks, it diverts the immune system and hijacks the cellular mechanisms to make itself more and more. And once it becomes more and more, it starts defending the immune system. That's how
Thank
we
you, sir.
get the disease.
Thank you, sir.
Good morning, sir.
Good morning.
Yeah, my name is Dr. Gamji, sir, from CSIR Mary Nagpur.
Nice to meet you.
I am also old student of Usman University Department of Zoology.
Good to know.
Sir, I have the two doubts.
Sure, please go ahead.
Yeah, one is, sir, you said the immune surveillance. The one
Can you repeat that, please?
said,
Can you speak a little loud and repeat the question?
Sir, my voice is audible?
Yes.
Yeah, sir, you said the immune surveillance.
Immune surveillance.
You mentioned the accessory proteins in immune surveillance. You can please say the accessory protein specific name?
Yeah, the accessory proteins for see, this is a 6 months old or 10 months old virus, and even those coronaviruses are there from past many years. We're still trying to figure out what accessory proteins does. And it's the names are, the naming is given like 3B, 3A, such as once the function is known, it will be named on based on, like, for example, NSP12 is one of the non-structural protein. Its function is replication. So that's why it is named as a replicase. But accessory proteins were not, their functions are not clearly known. That's why their names are like 3B, 3A, 7A, 7B, the numbers and alphabets. I hope I answered your question.
Yeah, another question, sir.
Please go ahead.
Yeah, mutation is depend on age. The mutation is most common in people of which age?
Mutation is most common in people of which age?
Yeah.
Is that your question?
It depend on age.
Do you mean the virus? Do you mean virus mutation?
Yeah, coronavirus.
No, it doesn't matter. Virus doesn't matter. Viruses constantly mutates as long as it has a human cell. Doesn't matter with the age or
depend on age.
Male or female or anything else.
Okay. Thank you, sir. Thank you.
Hello, sir.
You're welcome. Sure.
Can I ask one doubt, sir?
Please go ahead.
Sir, I am from SR BGNR Degree College, mathematics lecturer. So my doubt is, nice to meet you, sir. My doubt is, so if coronavirus, a single particle of coronavirus enters human body, it replicates more and more. It is true. So then, what is the relation between viral load and infection? So some people are saying that less viral load does not lead to infection. More viral load leads to infection. What is the relation between? Is there any relation or not? Whether?
Yeah, there is a very good question. Very good question. Yeah, the low viral load, high viral load, right? So there is always, see, one virus is, when I said one virus is sufficient to infect the human body, means if it escapes the immune system, it will start making more and more, and it becomes more viral load, right?
Okay.
And there is always a fight going on between the immune system and viruses. In the fight, if the immune system wins, virus dies, clears out from the body, and you don't get the disease. If the virus wins, then it takes over the body and you start the symptoms, and then it will go to the mild, medium, severe, right? In the severe stage, you have a high viral load. So when the viral load is low, immune system is successfully fighting, trying to push it down, basically, the viral population. That means immune system is working. That's why the young people actively fight, the immune system in the young people actively fights with the viruses and puts it low and clears them out from the body. That's why even though without any hospital or medical treatment, they can cure themselves. I hope I answered the question.
Okay, sir. Thank you, sir. Thank you.
Hello. Good morning, sir.
Good morning. Good morning, sir.
This is Anil Kumar, sir, Assistant Professor of Botany, Government Degree College, Manugur.
Nice to meet you.
I just want to, I have a small query, sir, with continuation of whatever you told. Once the virus it subsides in our body, so after getting infection and okay, after taking all this medication, once it subsides, so what about the inflammatory reactions in our body? And so I'm just talking about a long COVID.
Yeah.
So are
Excellent.
we ready for that? And what are the future implications of that? Can you throw some light on it?
Very good question. Excellent question. So the SARS coronavirus 2 is, well, again, I just mentioned the 10-month-old virus. Long-term effect on the human organs or human body or human inflammatory systems, not clearly known. Very few, handful of literature is available. Recently, I have read that cardiac system is being affected, but how much, how well, it's only like a few 30, 40 patients are being studied. We need more data from more patients to get a good conclusion how well it's being impacting. But very initial stage, initial result, we know that is it's clearly creating a cytokine storm, that too pro-inflammatory cytokines, which are damaging the organs. That is leading to the death. Organ damage, tissue damage, organ malfunction, leading to the death, right? So even though some people recovered, there may be, again, some damage may be already there, but body recovers with the time, with the habits, right?
Sir, and one more thing, sir, whatever these pharma companies, pharma giants like AstraZeneca and Pfizer, so I'm reading in newspapers that there are some patient is died due to failure of this coronavirus vaccine, like that in clinical trials. So
the thing is, it's an accident. So accidents happens, so in the clinical trials, there are two methods called systemic reaction, serious adverse events. So when a person dies, it's called a serious adverse event. That's why immediately the vaccine, the trial will be stopped or paused immediately. It will not move forward. And they will try to figure out why and what's happening in there. So in Johnson & Johnson case, they figured out that he had another complication. He did not die because of the virus vaccine. He died. He died because of the other complication. And interestingly, he was a placebo candidate. He was a control candidate. He was not given a vaccine. He's a control, so he was given a placebo to compare with the vaccine guy. So clearly, it states that it's not because of the vaccine.
Sir, okay, sir.
Good morning, sir. This is Srilata from.
Good morning, ma'am.
This is Srilata from Government Degree College, Bellampalli, sir. Sir, I heard that persons with O positive group are less prone to coronavirus. How far is it?
That's somewhat true. That's somewhat true. How, why is not quite clearly known yet. In the beginning, there are some reports saying that it's again population study, right? So let's say you take 1,000 patients who infected with coronavirus, and then find their blood groups. Most of them are AB or B or something else, 0 O positives. And then, so one laboratory study was done saying that the O positive blood group patients or persons are kind of ineffective towards these COVID-19 infections. But there's some literature is there. Yes. Go ahead. So somebody's asking.
Dear participants, if you.
So I can read the chat questions. So Mr. Mansur Ali is asking that what about herd immunity? happy to say that children are not affected corona compared to adults. Can we extract this specific power from children? So herd immunity is a concept. It's not very well proved yet. Herd immunity is basically like, once the virus comes into everybody and once it is gone, everybody develops the antibodies, memory cells, so that it will not come back. That's the concept of herd immunity. Without a vaccine, without medications, it is not possible in a large population, countries with large populations. So you cannot extract these things. This is just, it's a theoretical concept. So you cannot extract that from the children or anybody else. Is it necessary to take flu shots in the US against coronavirus? Flu shot is for the flu virus. It's not for the coronavirus. So coronavirus, there is no vaccine yet as approved. So the flu shot is for the flu. Flu virus is a seasonal virus, right? Not only US, anywhere in the world. What is the parameter to measure the coronavirus load? Yeah, in the laboratory, we do tissue culture infectivity dose. That's what is it called, TCID. Virus particles will be measured. There are various different measures are there. And I'm trying to read the chat box. Give me a minute. Go ahead.
Sir, once again, sir. Good morning, sir.
Good morning.
Can you create a vaccine from the SARS virus vaccine? Is there any possibility? Then because you are working upon that.
Yeah. Oh, you mean me? Can I or can anybody?
Yeah, especially we expect a vaccine from you, sir, because you are an Usmanian. You are Usmanian.
Oh,
you are
thank you so
and
much.
we are very obliged and very kind to see you, sir. We are expecting a vaccine from you, sir. Great legendary. I am very happy to see you, sir.
Thank you so much.
It was very well presentation, sir. Thank you, sir.
Thank you. But see, unfortunately, I'm not working on a vaccine. I'm developing antibodies. But our Bharat Biotech is working on developing a vaccine using the inactivated coronavirus. So not only me, there are so many different companies, various people are working on this. But I'm hoping that, I'm working on a treatment option, trying to develop an antibody, neutralizing antibody, so that we can treat people who already get the infection. So somehow, either a vaccine or a therapy, I'm contributing into this pandemic. I'm happy. And also, again, like being an Usmanian, also I'm happy. Usmania University student.
Thank you, sir. Thank you very much, sir. Thank you.
Sorry for breaking the enthusiasm of the participants to ask questions, considering time. Thank you very much, sir, for such an illuminating presentation. I express my heartfelt gratitude to you for joining us before 8:30 p.m. today. It shows your commitment. Thank you very much, sir.
My pleasure. Thank you so much for all the participants, and thank you so much for inviting me to address this webinar. So to the organizers, I would like to say one word. Dr. Ravi Shankar Garu, Dr. Srinivasulu Garu, and Principal Garu, and everybody, I may have to take a leave in few minutes because it's 1 a.m. for me in the morning. So I may not be able to stay until the end of the session. I apologize for that. But I certainly will follow up on the YouTube or something. YouTube, I'll continue watching later tomorrow. Thank you. Thank you so much. I'll stay for a few more minutes.
Sure, sir. Thank you, sir. Thank you, sir. It's my pleasure to invite Dr. Veerabrahmachari, Head Department of Biotechnology, Krishna University, to share his knowledge regarding the topic Pandemic Evolutionary Journey of SARS-CoV-2 from Viral Genes to Clones. I request Mr. G. Srinivas Kumar, Assistant Professor of Zoology, to reveal the detailed profile of the resource person.
Very good morning to all. We have a great resource person with us, and I'm exhilarated to invite Dr. Pallaval Veerabrahmachari, sir. He is currently a senior faculty member of the Department of Biotechnology at Krishna University. He has benevolently accepted our invitation to share his knowledge despite of his hectic schedule. Earlier, he served as postdoctoral research scientist at the Bacterial Pathogenesis Laboratory in Queensland Institute of Medical Research, Brisbane, Australia. He was also a DBT postdoctoral fellow at the Department of Microbiology and Cell Biology, Indian Institute of Science, Bangalore. He completed his PhD in microbiology from Goa University, India. His major research interests are the investigation on the applied research in microbiology and cell biology, including biochemical mechanism of bacteriobiofilm formations, structure-function relationship of bacterial rhamnolipids, and basic and translational cancer research. He published more than 125 research articles, including 5 international books, in prestigious peer-reviewed international Scopus indexed journals. He served as the editorial board member and as a reviewer for a number of national and international journals. He is a member of many international scientific societies and organizations. Most importantly, Indian Science Congress and Society of Biological Chemists. He has successfully completed 2 major research projects. He was awarded travel scholarship from QAMR, Australia, for attending 4th Indo-Australian Biotechnology Conference at Brisbane, Australia. He was also awarded with Young Scientist Travel Fellowship from the DST, Government of India, for attending 17th Lansfield International Symposium at Porto Heli, Greece. Dr. Brahmachari sir was conferred with various prestigious awards, notably Science Education Research Board, Government of India Young Scientist Award. He was nominated as Associate Fellow of Andhra Pradesh Academy of Sciences. He was a master trainer in Andhra Pradesh for English Communication Skills Project by British Council and APSCHE. He got Andhra Pradesh State Best Scientist Award in 2017 and Dr. V. Ramalingaswamy Memorial Award for Biomedical Sciences by Science City of Andhra Pradesh. He also obtained 2 Indian patents in 2017. When it comes to his experience, he has more than 13 years of teaching and research experience at the university level. With this brief introduction, sir, I request Dr. Veer Brahmachari sir to address our participants. Thank you, sir.
Can you all see me? Hello? Am I audible to all of you?
Yes, sir.
Yes, sir.
Yes, sir.
Shall I go on the full screen? Yeah? Fine.
Yes, sir.
Okay, fine. Very good morning to all of you, and I'm profusely thankful to the principal of the college and the convener of the seminar, and my good friend, Dr. Ravi Shankar, whom we spent a quality time at Usmania University for 1 year, and the department colleagues of Dr. Ravi Shankar, zoology, botany, and all the departments of the college and allied colleges as well. And also wonderful speakers, especially Mahendra Diwal has wonderfully explained about the vaccine strategies, and other people are also there. And definitely, I'm pretty sure today it's going to be a very great, enlightening lecture for all the people listening today. And shall I go ahead? Yeah.
Yes, sir.
Okay.
Yes, sir.
Okay, fine. Fine. Today I'll be talking about. I just have got 1 hour, 1 hour time, and I need to rush up. Okay. So today I'm talking about SARS-CoV-2 pandemic evolutionary journey from genes to clones. So I'll talk about this title at a later stage, and this is the structure of a virus which I've depicted in the cover page, and this is how the finally vaccine strategies are there. And I'm pretty sure today my lecture will limit only up to evolutionary journey only, and why RNA virus, why not DNA virus, why RNA virus is ruling, and why can't we prepare vaccine for such kind of viruses like SARS-CoV-2 and SARS-CoV-1. Okay, so there are several obstacles. We'll talk about them definitely in the coming slides. So let me start from the beginning, because we will say where it has started and how it has gone, and where are we now, and how are we tackling the pandemic situation, and where exactly this evolution has taken place, and where will it end. Okay, we'll talk about all this thing. I think it's a general information to everybody. We will talk about the involvement and role of bats and pangolins and humans, and how there is a lot of sharing of genes, deletions, insertions, and everything are taken place. Okay, so when you look into the history of pandemics, I think Mahendra Diwal has spoken pretty well about the vaccine strategies, but we'll talk right from the beginning onwards. Okay, so we have seen several pandemics so far, and we are talking about the very small dot staying at the below. Okay, novel coronavirus is 7.9K. It was a old data, but however, if you look into the Spanish flu, Russian flu, and Hong Kong flu, Asian flu, swine flu, there are so many smallpox and bubonic plague. So many millions of people have died, and okay, so it has throughout the history it says that it has spread across the world, and infectious diseases have been a constant companion with the human beings. And even in this modern era, outbreaks are nearly very constant. And here are some of the history's most deadly pandemics. Okay, one among them is novel coronavirus. So if you look into the outbreak story, it's the deadliest pandemic history. You can see here, okay, the measles 200 million, Black Death, bubonic plague 75 million, smallpox nearly 300 plus million, Spanish flu 50 to 100 million. We are just talking about 1 or 2, 3 or millions. Okay, just look at here. There's so many big things like typhus, cholera, Hong Kong flu, the third pandemic, second pandemic. We have seen so many of them, but we are still we need to see so many RNA viruses in the coming future. Okay, we have seen only one. Okay, we may have to see some more RNA viruses in the coming years. Okay, so where did it start exactly? So it's basically a zoonotic virus. Okay, it's basically the transmission took place from animal to humans, the emergence of a new virus. Okay, it took place in 5 stages. Look at here. It's basically a zoonotic virus, basically belongs to coronavirus. Okay, and then in stage 2, it has, okay, and then it has become a animal disease reservoir where the virus naturally lives in bats. For example, for Ebola and SARS also lives in the bats. Okay, and the stage 3, there is a species barrier, the jumping from one animal to another animal. So being one of them as an intermediate host. Intermediate host in the case of SARS, especially civet cats, pangolins, and okay, so they made a major role in in acting as intermediate host. And then in stage 4, transmission to humans, then between humans, and then the virus became adapted and then started mutating. And in the stage 5 dissemination, we almost crossed the stage 5. This is dissemination is aided by increasingly mobile populations. So this is how it exactly depicted the stage from animal to animal transmission, and animal to human transmission, and human to human, human to human, human transmission. Okay, so that is how the dissemination has taken place. So if you look into broadly, this coronavirus is basically belongs to coronaviridae. Okay, it's again divided into 4 types: alpha coronaviridae, beta coronaviridae, gamma, and delta. Okay, the coronaviridae which we are talking about now, it's beta coronaviridae, and it comes into the lineage A. Look at here. These bats and these avians, okay, and from here it has moved to interspecies transmission from bats to another bat species, okay, and from bats it has moved to animals, other animals like intraspecies, okay, interspecies. This is intraspecies, interspecies transmission, and from here animal to human transmission, and from here human to human transmission. This is how it basically looks like. So this is where the exact thing, okay, the lineages B, C, D, all the merds and other things comes under BCD, but the one which we are talking about is comes into lineage A. Okay, so if you look into here, the primary host is mm, it's bat. Okay, the phylogenetically and genomically they are most similar to SARS, like bat cover coronavirus. Okay, so people have consuming were consuming bats as a food in China. Okay, and then with the help of intermediary host and with the help of direct transmission, humans picked up the infection, and from there human to human transmission had taken place. And now people are looking out for therapeutic strategies. I'm pretty sure all these remdesivir, lopinavir, and all these are not exactly responding to the individuals, especially in Maharashtra and Tamil Nadu and even Andhra Pradesh as well. So if you look into the SARS and MERS, what are these SARS and MERS? Who are these MERS and SARS? I'll talk about that. Okay, SARS and MERS both are closely related to bat strains of coronavirus. They're transmitted through other secondarily infected species, especially SARS Himalayan palm civet cats, and MERS are dromedaries. Dromedaries are nothing but camels. Okay, so SARS originally isolated with wet market in Guangzhou. It is moved to more than 26 countries, and a lot of economic damage has taken place. More than 50 to 70 billion dollars of money has been gone and down in the drain. And look at here, this is SARS and this is SARS-CoV-1 and SARS-CoV-2. Okay, and this is MERS. Look at here. Okay, though if you look into the clades, the evolutionarily, okay, so these genomic characterization and the epidemiology of 2009 novel coronavirus, this has a lot of implications for viral origins and receptor binding based on the ACE2 receptor. Okay, we'll talk about that at a later stage. So I'm just trying to show you the distance between the SARS and the MERS and the SARS and SARS-CoV-2 and SARS-CoV-1. Okay. So bats, I think zoology people must make a note of this slide. Basically, why bats? Why bats are making a lot of involvement, bat as a reservoir as emerging viruses? Because bats are having varied food choices, colonial, they are solitary or nature, solitary in nature, and population structure, it's very diverse, and they can, the ability to fly, seasonal migration and daily movement patterns and lifespan of bats is pretty high compared to other avian mammals, and roosting behavior, ability to echolocation, they can echolocate from one place to another very quickly, very fastly, and viral susceptibility at a time, the bat can host up to nearly 500 to 700 viruses being asymptomatic. Okay, this is a great part of the virus. And bats are found on all the continents except Antarctica. Antarctica, you don't find bats at all. And viruses isolated from infected bats worldwide so far. Rabies, Lissa virus, influenza, Hendra, Nipah. Nipah we have seen in Kerala recently. And corona, Japanese encephalitis virus, chikungunya. Okay, all these are found, come out as outbreaks from the bats. And if you look at the importance, the benefits, beneficial part of the bats, they help control insects, they reseed cut forests, they pollinate the plants, balanography, balanophily. Okay, their guano, okay, their excreta are used as a fertilizer. They are models for sonar systems, echolocations, okay, because they can detect anything through sound or sonar navigation. Okay, so bats, why are these bats are reservoirs of so many deadly viruses? Why? The reason is the bats have difference in innate immunity, okay, because they have a lot of differences in their innate immunity, unlike we don't have such things. And there's a lot of constitutive production of interferon gamma, interferon production, and decreased or absence of pyrin, HIN, domain proteins are there in the innate response proteins in the bat immune system. The viral coevolution, part of a bat gut virome, okay? So this flight while flying, the flight results in increased ROS production and the body temperature. Always the ROS production is pretty high because compared to other human beings and other animals. Viruses may have also adapted to the higher temperatures. Okay, so these viruses can adapt to very high temperatures compared to other avian mammals. So most of the time, bats don't get sick. They don't. There is a noise. The coordinator, please take care of that. Okay, bats don't get sick at all for three reasons. Evolutionarily speaking, it's not advantageous for the virus to kill a large population of its virus, of its host. The viruses depends on them to survive and to continue spreading onto other animals. Unlike in human beings, bats' immune system fights the viral infections pretty well, but does not overreact to them, which protects bats from the deadly hyperinflammation. Because we see the hyperinflammation in humans, but we don't see the hyperinflammation at all in bats. And moreover, these bats have got a constitutive reactive oxygen species production continuously happening. And as the time passes, the virus becomes less lethal to the original host. That is what has happened in bats. So now it's become attenuated to the bats and pangolins, and now that particular part where the deletions and insertions have occurred, so that particular part is really making a lot of damage to human beings and really creating pandemic kind of situations across the world. So because of this jumping, this virus has got a habit of jumping between species and jumping beyond the species. So that is where if a virus jumps into new species again, apart from pangolin or civet cat or a bat, okay, if it finds a new host and comes back to humans again, then it will be not in at least in lakhs, it will be in billions and trillions. It will even cross the smallpox or bubonic plagues and Hong Kong and Spanish flus as well. Okay, so if you closely look into the similarity, the evolution, the genome part of it, COVID-1 has come out in 2002, okay, and COVID-2 has come out in 2019, and in between MERS has come out in 2012. All belong to beta coronavirus, okay. And look at here the reservoirs for all three of them. COVID-1 is civet cats, and COVID-2 is pangolins. It's still a question mark. It's a pangolin or a bat. But for all three of them, the common ancestor is bats. Look at here, okay? And for MERS, it is camel. And all three cousins, okay, have derived their genome from the bat, okay? So COVID spill over to humans from here. It has taken place. And if you look into COVID-1, the death rates and okay, the pandemics and are pretty low compared to, because it does not have a pandemic kind of a genome spillover when it spilled over to human beings. And when you look into COVID-2, it's really creating a lot of pandemic. And recently, yesterday's news in India, Kerala and Maharashtra again it's turning back with the new symptoms and new infectious ratios and okay, so it's again getting a problem in November and December, probably in Kerala and Maharashtra. And when you look into MERS COVID, the deaths are pretty less compared to, so it's come in Middle Eastern countries, Gulf countries, okay, so Eastern Africa. Okay, so this is where it has come out as an outbreak, and very soon they could contain the viral capabilities in that area. So if you look into here, the host body environment and the external environment, what's happening here? Look at here. The bats is the ancestral viral reservoir, and this intermediary host, the pangolins, the individual transmission, then silent carriers and mild infections, and susceptible hosts are the people who are targeted finally. And here, look at here, the species spillover. There is a lot of selective pressure occurring between the bats, pangolins, and civet cats and other animals. Then a lot of mutations, recombinations are taking place. The continuous molecular genetic evolution of new viral species every now and then. I'm pretty sure only in India it has crossed not less than 60 to 70 serotypes with a lot of mutations and recombinations have taken place, and new subtypes and new variants and new serotypes are generating every day by day. And finally adaptations inside the human host body. People are saying that in Indians especially, basically we have contained a lot of melanin in our human skin, and on top of that most of the Indians have got hnRNA in their human cells. So for that reason we are recovering pretty faster compared to white people, okay? So the natural selection by the host, the infection establishment, then host susceptibility, high transmissibility, okay? So these are the other reasons why it is pushing COVID-19 to global pandemic and making havoc across the country and across the world as well, okay? And through these respiratory secretions, through droplets, aerosols, and removing the viruses, there's a shielding effect. There are a lot of environmental effects also, temperature, relative humidity. Okay, people are saying that temperature does play a role, and sometimes people even say that temperature does not even play a role, because if temperature had really played a role, I'm pretty sure, I think in the last summer we must not have got infected with coronavirus in India at least. Okay, so I'm pretty sure this temperature factor and humidity factor might vary from individual to individual, person to person, region to region, and their immune system, immune susceptibility as well. So if you look into here, the origins, look at here, SARS-CoV-1, the origin is for all three of them, the origin is bat coronavirus, okay? But if we coronavirus 2, COVID-2, if you look into here, RATG13 region has been inserted in this particular thing. So this differs from MERS-CoV and SARS-CoV, although they belong to same viral clade. Okay, it has moved to civet cat to humans and camel to humans, and pangolin could be the origin to humans. And the whole genome of COVID-2 is aligned with the genomes of all other three viruses, BATS-CoV and CoV RATG13, in Rhinolophus affinis species, horseshoe bats of Yunnan province with 96% similarity. Okay, and COVID-19 virus likely emerged from recombination of viral genes across the different species. So COVID-2 has, what people are saying is it has become a recombination of genes across different species. That is why this has become a virulent strain and virulent serotype, which can infect a variety of species, okay, and variety of organs and variety of cell lines, variety of tissues, okay. So this kind of peculiar system was not seen in any other RNA viruses before. Okay, so pangolins, why pangolins may serve as intermediate hosts? Pangolin's whole genome had 91.02 similarity with SARS-CoV-2, this 91% similarity, and 90.5% similarity with bat COVID RT-G13. Okay, it's a recent report in 2020, current biology. Then emergence of SARS-CoV-2 through recombination and strong purifying selection pressures. Okay, then SARS-CoV-2's entire receptor binding motif, RBM, receptor binding motif, was introduced through recombination with the coronavirus from pangolins, possibly a critical step in the evolution of SARS-CoV. See, this picking up a gene from all the species and putting up in the receptor binding motif, especially in the pangolins, okay, could be the major critical step in the evolution of SARS-CoV-2's ability to infect human beings. So this is one of the major milestones in the evolution, especially in deletions, insertions, and recombinations that took place in this RNA virus. Is really going to create a milestone in the history of evolutionary viral history. Okay, so if you look into hop, skip, and jump, hopping, skipping, jumping, probably the frogs will do that. Okay, jumping, hopping, skipping. Okay, so if you look into the genomes, these are the common respiratory coronaviruses have long circulated between the human beings. They're all close relatives to each other. Okay, and look at here, mouse, rat, vole, the camel is the intermediary host for MERS. The lethal coronavirus have jumped into humans 3 times. Okay, MERS have jumped only 3 times, whereas COVID-1 has jumped only once, and COVID-2 is still jumping. Okay, it's still jumping, and MERS has stopped, COVID-1 has stopped. Okay, and for all of them, there are common ancestors. Okay, all have been circulated among them. See, previously these genomes were circulating, these serotypes were circulating, and now it's pretty well that SARS-CoV-2 is the only major player in the history, evolutionary history. Okay, so if you look at the potential origin of SARS-CoV-2, okay, so if you look into the origin, okay, so the SARS-CoV-2, COVID-1, okay, from bats to civet cats to humans, okay, so this is if you look into this part, okay, it's low to moderate, okay, and COVID-2 from bats to pangolins to humans, and then human to human transmission. So this is red line determines a pretty high kind of back-to-back transmission, okay, and MERS-CoV-2 also, look at here, this is comparatively less compared to SARS-CoV-1. Okay, so the very basic title, genes to clones, clonal expansion, clonal diversification, has as a basic overview from my title, saying that there is a lot of variations occurring between genes and proteins in the RNA virus. Okay, so why there is a clonal expansion? See, it has started here as an yellow and slowly mutating in one nucleotide, mutating another nucleotide. See, look at it, all the started in China as a yellow spot and moved to East Asia in all the orange spots, USA in all the blue spots, and Europe in all the light green, and Australia and all the violet, and Mexico and Brazil all the green spots. Look at here, even it has gone to very far off places, and node A is a cluster. From that cluster with a single nucleotide mutation, single nucleotide polymorphism. Okay, so it has recombined and obtained a new serotype in the form of RT-G13 from Yunnan province. Okay, so these circle areas are proportional to the number of taxa, and each notch on the links represents a mutated nucleotide position here. Okay, so if you look into the potential origin, so there are 3 clusters here. Okay, cluster A is ancestral type. Okay, and cluster B is derived. Okay, all cluster B is derived from cluster A via 2 mutations observed to mutate slowly in China but rapidly outside China. Okay, and this cluster A is the closest to the coronavirus found in the bats and pangolins, considered to the root of the outbreak. Have 2 subclusters, Wuhan cluster, and another cluster is America and Australia cluster. And cluster C, okay, daughter of cluster B, one mutation different from the parent variation spread to Europe via Singapore. So from here, from Europe to Singapore to Asia, okay, we fall in cluster C. Okay, so if you look into the basic the difference between pangolin, COVID-2, bat RAT-G13, and pangolin. Look at here, the lineage, okay, the similarity, the phylogenetic evolutionary similarity. Okay, so this lineage apparently lost a specialized receptor binding domain that helps the SARS-CoV-2 infect the human cells. Okay, this lineage has lost the receptor binding domain. So, and there is an insertion of bat RAT-G13 in SARS-CoV-2 and a part of gene from pangolin. Okay, so this is now unable to bind to the receptor domain of their own cell line, and now it is able to infect the human cell line pretty well. So I'm trying to show how close they are and how distant related they are, how much close symptoms they are. Okay. So if you look into the past, present, and the future of coronavirus evolution, so what evolution must have led to SARS-CoV-2's ability to spread in humans and to cause pandemic? This is one of the major questions. So what evolution must have led to that, and how is SARS-CoV-2 evolving during the pandemic, and what future evolution could be problematic? Should we prepare for it or not? I was talking about the, again, one more species jump from one animal, especially the bat, to a pangolin to other animal and back to human beings. That will be a major problem in the coming days. Okay, so yeah, see, let me show you here RAT-G13 and pangolin genome. See, some part of pangolin genome has been inserted here in SARS-CoV-2. So this is really playing a major role in the recombination, and especially that has taken place only in the S spike protein only. So the recombination that has enhanced the spike protein interactions with the human ACE2. So this is where, look at here, there are 4 mutations have taken place in the bat. Okay, and look at here, in the humans, little variations, and in pangolin only, okay, and only 5 mutations here. So from COVID-2 to RAT-G13, okay. So for COVID-2, RAT-G13 is much more closely related than pangolin, okay. So if we look into the new study, one of the new study found that you take any part of the spike genome. There are 4 regions in the spike protein. Okay, you take any region and try to look out their evolutionary history. So every region in the spike protein has got, okay, different evolutionary history. It can interact with any number of cell lines. Okay, this indicates that different segments of the genome have different evolutionary histories and indication of recombination. So every segment can undergo recombination thousands of times. So that is where it's a major problem. It has come in recently, 2020, scientific advances. Okay, so especially if you look into the genome structure, the mutation is taking place very, very rapidly. Okay, so this genome is relatively compared to other viruses, this genome can pretty well recombine and mutate much more fastly compared to other viruses. Okay, so why it is every time RNA viruses causing global outbreaks, and why not DNA viruses? What's happening in RNA viruses? Why RNA? It's a big question. Why RNA viruses are mostly involved in all the outbreaks? So there are 180 recognized species of RNA virus that can infect humans, and 2 new species are added every year due to recombinations and mutations. So a big question mark is why RNA viruses? Look at here. If you look at here, reason 1, RNA viruses mutates literally 1,000 times more than DNA viruses and due to lack of proofreading capacity in the RNA polymerase. RNA polymerase lacks a RNA proofreading capabilities. Only DNA polymerase has got a proofreading capabilities, and mutation rate in RNA virus is quite high compared to DNA viruses. Okay, so that is the reason why, is one reason why. Then RNA viruses mutates once every 50,000 base pairs per infection. Okay, not per outbreak, per infection. So each time it infects, it can keep on mutating within 50,000 base pairs, and DNA viruses mutates once every 5 billion base pairs per infection. So this is compared to nearly 1,000 time dilution. Okay, and more mutations means there is less chance of vaccines to be effective. So I'm pretty sure preparing a vaccine for RNA virus, especially SARS-CoV-2, is not that easy, because more mutations, because by the time you prepare a vaccine, this vaccine serotype may come out into the environment and interacts with the wild subtype, and even emerges in itself into a new subtype, a new serotype, and the less chances of vaccines to be effective against RNA viruses compared to DNA viruses. Okay. The reason too, RNA viruses violate the central dogma. Central dogma, you understand pretty well. DNA to RNA, first initially replication, DNA to RNA is transcription, and RNA to protein is translation. But this rule does not apply to RNA viruses because it violates the central dogma, and this is a naturally gifted potential which they possess. And now, what is central dogma of biology? If you look at the say, I told you. DNA to RNA, RNA to protein. So this strategy doesn't work with RNA viruses, especially because it's completely away from all these rules. And so majority of the RNA viruses has the luxury to make proteins in its own style directly from their RNA genome when it just enters into the cell. So the soon after it enters into the gel, it can start preparing its RNA proteins, RNA genes. Okay, so viral mRNAs, and that's why they can easily cause infections upon entry compared to DNA viruses, who has to follow 3 steps: replication, transcription, translation. All this blah 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 story will not be there in RNA viruses. And reason 3, all the RNA viruses are mostly transmitted from animal to human transmission. That's zoonotic transmission. Almost 89% of infections caused by these viruses come from animals. So look at the bats in the background. So they hang upside down, and mostly they are metabolically very their what do you say, metabolic index is pretty high compared to even human beings and even rats also. Okay, so just recall the origin of these infamous viruses like HIV, AIDS, influenza, dengue, Ebola, chikungunya, Zika, corona, Nipah. All of them came from animal to human transmission. So this is the major reason why we are very much closely related to animal viruses. And reason 4, all the RNA viruses are small in size, especially 20 to 200 nanometers compared to DNA virus. DNA virus is comparatively pretty higher in size, like 100 to 800 nanometers, thus providing the few epitopes displayed in the immune system. And RNA viruses are often without outer membranes, envelopes, okay, and provide resistance against disinfectants and sanitizers. So most of the sanitizers sometimes does not work with RNA viruses because they does not have an envelope on top of them. So there's a major drawback with RNA viruses. Okay, and due to small genome, they can also reproduce much faster. They can and quickly evade the immune system by forming new strains. Diversification or infections pretty faster compared to other things. And reason 5, DNA viruses replicate in the nucleus and have to follow the rules. Okay, follow the same rules like its genes, while RNA virus replicates in the cytoplasm. So that's the main reason. As soon as the RNA virus enters into the cell, it starts preparing its viral proteins. And whereas DNA has to go into the nucleus, then prepare, unwind itself, and then do all kind of very lengthy procedures. Okay, so RNA viruses easily expresses its genes to perform any kind of infection. And in DNA viruses, the genome and the associated proteins to be transmitted to the cell and to the site of organ site of infection. Okay, that takes a lot of time. So that's about the basic evolution. And when you talk about contact tracing, India, okay, India is completely filled with a lot of traditional, cultural, historical, evolutionarily, okay. So we are all bound with a lot of feelings and emotions, and a lot of factors are there. So we can't close our borders appropriately. We couldn't do that because there's a lot of traditional, communal, and there are so many things involved. Okay, and a lot of socialized social stigma is there in the Indian population, especially. They don't want to come out and say that I've infected, and the ambulance comes to my doorstep and picks me up to the hospital, and people will think bad of me and bad of my caste and my honor and everything. Okay, only during the last stages of their life they come out and say that I'm infected. Okay, so there's a lot of problem, especially in India. Contact tracing is a very big problem. So we have led to tertiary, quaternary transmission, human-to-human transmission, aerosol transmission, community transmission. All this took place only because we could not do well. We could not spell out due to social stigma. So contact tracing was literally failed because a lot of revenue medical people were deployed in every town. They did a wonderful job, especially medical staff, especially doctors have done a wonderful job. But still, we left behind with a major gap in contact tracing. Okay, So let me not go into the contact tracing much. Okay, so generally we count the reproductive number, how a virus with a reproductive number R0, it's called as R0. Okay, it spreads from one patient to two patients, 2 to 4, and 4 to 16, 16 to 64, 64 to 256. That's how the transmission takes place. And if it crosses thousands, okay, contact tracing does not work at all. People will not reveal whom they have met, whom they have talked to. So the average number of people that one person with the virus infects based on R0 scale. So and for especially the R0 scale for COVID-2 is 2 to 2.5. Then in the month of March and April, now it has gone to nearly 4 or 5 in the R0 scale. The reproductive number, okay, it's keeping on increasing every day by day. Okay, so let me talk about the genomic epidemiology of coronaviral cases. Look at here. What exactly is India is such a big country, and we have got only one institute, like a National Institute of Virology, that's located in Maharashtra, Pune. And I'm not pretty sure that unlike other countries, like especially Netherlands, Belgium, and Scandinavian countries, Europe, USA, America, Australia, okay, all those countries, they sequence the genomes from every zone. See, depending upon the number of cases, they sequence the new varieties, new serotypes, new variants every week and submit to the central database, which is located in Geneva. Okay, and I'm pretty sure we are not doing this, and still the Indian country is not aware of from where the clone has started and which is a new variant, a new serotype, and how this transmission has been taking place. Can we control the one serotype which is majorly expanding, which there is a lot of clonal expansion in one particular strain? Okay, so this part we are unable to come out of it. So even if you prepare thousands of vaccines, okay, this doesn't work because we are technically not strong in terms of locating the clone and clonal expansion, serotype, a variant. And if there are so many cases in Maharashtra and Tamil Nadu, I'm pretty sure that must be belonging. See, that must be moving, crossing the reproductive number, and there must be a lot of mutations taking place. And only in Maharashtra and Tamil Nadu you might find not less than 60 to 70 serotypes. Okay, so that serotypes must be located, must be sequenced, must be submitted in the central database in order to know whether the already known mutations are taking place in the spike protein, or are there any new mutations coming up in the spike protein or not. So this is really pretty interesting thing, which Indian government is really failing in sequencing the genome and submitting the central database, where all other countries are doing pretty well in that. Okay, so if you do that, if you submit that sequence database, we can easily locate the genes, locate the clones. So which clone is really moving from China to Europe, and Europe to other countries, other countries to other countries. Okay, where it is moving to South Atlantic Ocean, South Pacific Ocean, okay, Madagascar or Brazil, or okay, Paraguay, South Africa, Madagascar, okay, Angola, Senegal, Cuba, Mexico, South America, Latin America. Okay, we can easily locate which clade it belongs to, which cluster it belongs to, which evolutionary variant does that belong to. We can easily locate in that way. Okay, so if you look into the variants, okay, it's called polybasic cleavage site. This is there in the ortho-linked glycan residue. Okay, so the binding behavior of the SARS-CoV on the O-linked glycan residue, look at here. So all that is taking place only in this region, PRRA. Okay, so if you look into the genome, this part is pretty much okay, variant. Okay, a lot of insertions taking place only in this part. So there are two major types, L type and S type of the circulating SARS-CoV-2 strain, and two major types. Okay, wait a minute. Okay, so look at here, two major types, L being the major type, okay, 70%, and S being the minor type. So we fall into the 30% minor type. And look at here, this region especially where I've shown, this is a genome. And this region, okay, the Asian population might undergo a lot of mutations. Let's look at here, L, S, L, 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 okay. So these genomes are going to be very much dangerous in the coming days, okay. So where there is, okay, yes, okay. So yes region is very much dangerous. Okay, so this is a picture taken from the BBC news channel. Okay, every second, every minute, okay, there was a update coming up from from China and other countries during the pandemic, that every minute, every second, the death toll was increasing. Okay, so recovery cases were pretty low and death toll was increasing like anything. So I wanted to show this picture because it was a minute-to-minute comparative study, okay, taken by BBC. Okay, so let me just talk about COVID-1 and COVID-2, how much they are similar, how much they are dissimilar. Okay, COVID-1 has come in 2002. The spill reservoir is known. This is civet cat. Still, COVID-2, the spill reservoir is unknown. The people are saying whether it is pangolin or bat. Okay, sometimes the people even said snake. Okay, the most transmission occurred in hospital only, but widespread community transmission and aerosol transmission, and no transmission until 24 to 36 hours of symptoms, and no asymptomatic cases. But whereas here, most of the cases are asymptomatic, we cannot even trace whether he is coughing or not coughing. Okay, greater affinity of SARS-CoV-2 for the host receptor, an increase in global travels, and because the increase in global travel has really taken a very drastic step in in terms of community transmission, especially in SARS-CoV. Okay, so wait a second. Okay, so next thing, if you look into the structure, okay, so it has got spike proteins and it has got RNA as a genetic material, and I've got envelope proteins, I've got membrane proteins. Okay, the genome is pretty smaller compared to, they have got just only 29 set of 29 genes, 29 coding genes. Okay, that means it can code up to only 29 proteins. Okay, so look at here the structure of spike proteins, viral proteins. Okay, so even a small change, a small nucleotide change in any of the protein, okay, in the spike protein. Okay, so all the green are structural proteins, and all the other colors are non-structural proteins, and yellow orange is your human proteins. Look at the interaction. Okay, so even if there is a small change in one nucleotide, okay, host viral complexes and even the vaccine serotype, vaccine epitope may not aptly go and bind into the receptor. Okay, so that's a major thing, and there's a wonderful structure. I've picked it up from the New England Journal of Medicine. Okay, look at here. It looks something like a big architecture. I don't think even IIT civil engineering and architecture can draw in such a way. See, envelope protein, membrane protein, ORF, open reading frames. Okay, single stranded RNA, nucleocapsids, NSP 13 helicase gene, NSP9 RNA replicase. Okay, and glycan groups, which goes and binds only on the glycan residues of the human cilia, epithelial. See, this is a human cell line which I'm showing. Okay, this is a human cell membrane with the human ACE2 receptor. Okay, class low. Okay. Okay, so human ACE2 receptor. Okay, so this is a human ACE2 receptor, it's a glycan groups and a spike protein, and okay, so it's a lot of things here. Okay, look at here, this is how the virus comes and binds to the ACE2 receptor. Okay, so if you look into the genome, okay, SARS-CoV-2 and MERS-CoV, okay, the genome just contains only 29 genes, and we have got 30,000 genes, and all the 20 out of 29 genes, 27 genes can interact with all 30,000 proteins and genes in the human system. So this kind of viral human interactions I have not seen and studied so far in my lifetime. So out of 29 viral genes, 27 genes have got the capability to interact with all 30,000 human genes and proteins. They're very peculiar. Okay. So if you look into the viral factors, especially S proteins and host factors, okay. So look at here, S1 region and S2 mediates a viral cell membrane fusion. Okay, so bat, swine, and civet. Look at here. So there are so many host factors which is involving. Okay, especially people above 65 years of age and people with underlying diseases, especially people. Okay, so who are having hypertension, chronic obstructive pulmonary disease, diabetes, cardiovascular disease. Okay, and severe complications. There is a disturbance in the background. Someone has switched on the speaker. Can you please hold them? Hello, coordinator. There is a noise background. I cannot speak. There is a noise background. Please unmute everybody. Hello. Hello.
Everybody, please unmute yourself.
Yeah.
Mute yourself. Somebody unmute, right?
Please,
Mute yourself.
there's a
Yes.
still voice coming up. Yeah, because I cannot speak in such case because yeah. Because I already crossed 1 hour time, and whenever you want, you can ask me to stop. I'm happy to stop anytime. Okay. So there are 16 non-structural proteins, NSP1 to NSP16. We'll talk about that, and especially severe complications like multiple organ failures and all these things will be seen. Okay. So the major story comes up in S1 and S2 because in the spike region, S1 determines the viral host range and cellular tropism by so RBD, receptor binding domain, and S2 mediates of viral cell membrane fusion by HR1, HR2. I'll talk about that at a later stage. So see, this is our throat region. Look at here. So this is our palate, tongue, and the part which I'm showing here. Okay, this is where the cilia or mucus layer is located. Okay, this is the mucus layer. So this is how the mucus layer looks like. Here I'm showing the cell membrane here. Look at here, human ACE2 receptor. Okay, this mucus layer I'm trying to show it in a most enlarged way. Look at here, our mucus layer, this thread like mucus layer is not that simple, that easy. Okay, there's a lot of so many layers, 1 2 3 4 5 6 7. Okay, only after the cartilage, connective tissue, and bronchial gland, smooth muscle, lamina proposia. Okay, only after everything you find your tissue. Okay, it has to cross so many barriers, the virus has to cross so many barriers. Only the coronavirus has got the capability to cleave with the help of molecular scissors, neuraminidase or hemagglutinin enzyme. Okay, and no other bacteria and no other virus so far has got the capability to cleave this mucus orthosialic acid linkage, especially in the throat itself. So people are saying that in the throat, in the epiglottis region, okay, in the cervical lymph node, especially here, this region, this virus can stay for 3 days. So if you feel like there is a hitch in your throat, okay, immediately go and gargle with salt water, okay, and hot water with the ginger and turmeric. Okay, if you do that continuously for 3 days, I don't think it will move down to the respiratory tract, lower respiratory tract. Okay, it will be expelled completely from outside the upper respiratory tract itself. Okay, if you look into here, so there is a viral entry. Look at here. There's a viral, viral assistant bacterial. Is not going. And this is not going from here. Okay, so viral assisted bacterial invasion in the lung. So what exactly happens is, this is a cilia which I showed here. This is the cilia which I'm showing. This is a cilia. Okay, these are all the layers. These are all the layers which I'm trying to show here. Okay, the mucous cilia. So virus acquisition takes place in the slide A. Okay, and neuraminidase hemagglutinin enzyme will comes and binds here and cleaves the sialic acids. Okay, and all the sialic acids and neuraminidase enzymes will come and interact here. Along with the viral acquisition, there are a lot of bacteria waiting outside the entry point to get an entry along with the virus. So I'm pretty sure it's not the virus which kills the human being. The bacterial pathobionts which come as a companion along with the virus, okay, which kill the human beings, because virus does its jobs, okay, pretty well, and once it finishes life cycle, it gets out of the body. Okay. However, okay, so when both are there, both are infecting continuously. Altered mucus, decreased mucociliary velocity, decreased AMP, antimicrobial protein presence, okay, and enhanced bacterial colonization, and finally it enters into the epithelium, endothelium, and then fibronectin, and then translocates in the tissue invasion. Once it enters into the tissue, that's gone. That means the person, if it enters in the tissue, that means next is blood invasion. Once it enters into the tissue, okay, septicemia, viremia, okay, bacteremia, or septic shock syndrome, all these will be undergone. Okay, and look at here, this virus, and this is the cilia which I'm trying to show. All this virus will go and bind to the cilia this way. Okay, and first time in the history, I think I told you before, or I'm not, I told you before also. Look at here, and this orthosialic acids, the mucus layer produced, which is present in the throat. Okay, this is the only sialic acids, okay, only carbohydrate moieties produced inside the nucleus but not in the cytoplasm. You take any plant carbohydrates, animal carbohydrates, human carbohydrates, okay, yeast carbohydrates, you take any carbohydrates in the globe, all the carbohydrates are biosynthesized in the cytoplasm itself. But this is the first biomolecule, orthosialic acid carbohydrate conjugate moiety, which translocates inside the nucleus, and inside the nucleus, okay, it gets biosynthesized. Okay, see, there are glucose moieties in the cytoplasm with the help of N-acetyl glucosamine and N-acetyl mannose, N-acetyl mannose 6 phosphate, neuramidic acid. Okay, with the help of sialic acids. Okay, it moves inside, and with the help of all the sialic acids, once after synthesis, it moves out of the nucleus and gets post-translational and glycosylation modification takes place in the cytoplasm with the help of Golgi complex and endoplasmic reticulum. Look at here. So all these modifications takes place in the cytoplasm, enters into the nucleus. Inside the nucleus, this orthosialic acid is synthesized and comes out of the nucleus through nuclear pore, and with the help of silyl transferase enzymes, okay, it gets modified on the Golgi apparatus, and from there, okay, that means what I'm trying to say, we have got N-linked sialoglycans, O-linked sialoglycans, and gangliosides, and this O-linked sialoglycan is the one which is trying to stop all the viruses and bacteria. So if we don't have O-linked sialic acid on our mucus layer, okay, that means we will be much more prone to all the viral infections, because coronavirus, all the coronavirus family, all the viruses can cleave orthosialic acids with much ease and much simplicity. Okay, so I'm talking about the barged entry of bacteria. Okay, look at here, barged entry, commensal versus pathogenic virus. We look into any part of the human organs, especially mouth, pharynx, respiratory system. There are so many bacteria waiting outside to get an entry along with the virus. Okay, and you talk about skin. Okay, skin. These are the bacteria which are trying to enter along with the skin, and urogenital tract, because these are all the openings where, okay, even stomach. There's so many bacteria. Okay, they go and live along. Come along with the virus and live in the stomach and intestines and create a lot of mess. Okay, these are the invasive bacteria, the very dangerous bacteria comes along with the virus. Okay, they exert a lot of virulence factors, a lot of host invasion, and cause disease, evade in host defenses. So they can evade any immune system, come along with the virus. Okay, and see, this is how the host invasion strategies. This is a mucous ciliary layer. Okay, this is how the bacteria will go and bind to the ciliary layer. So initially, how do they colonize? How does the bacteria colonize along with when they come along with the virus? Okay, initially they come as a pioneers. Okay, and slowly they become settlers, just like how we go and settle in America, and slowly they become a society, and slowly they become a community. Once they become a community, that means this community is going to kill a human being. So with the help of cell signaling, co-aggregation, metabolic synergy, genetic exchanges, a lot of gene transfers, lateral gene transfers, horizontal gene transfers. Okay, each stage in the colonization may promote the tissue invasion and death finally. Okay, so I'm pretty sure not only the virus, so bacteria coming along with the virus also can kill the human being. Okay, so we are thinking that only virus is killing the human being. So this part is a second part. So this part is a part of after this part. See, this is A. Okay, and B is this one. Look at here, the B. Once it entered into the tissue, then after the tissue is bloodstream. Okay, once it entered the tissue, all the cells, macrophages, dendritic cells, interferons, all the cells will start coming to the site of action of virus, because all the green particles are viruses. Okay, so there's altered cytokine response, interleukins, neutrophil recruitments, okay, and altered TLR pathways, impaired natural killer cells, so neutrophils, bacterial proliferation. There's so many things happening once the virus gets inside. That means, so apoptosis will be there happening. Okay, once enters into the bloodstream. Okay, and that means once enters in the bloodstream, that means the human being is absolutely no more, because if the virus and bacteria both together enters into the bloodstream, that means the human being will die for sure. So all the people who survived so far from the hospitals are recovered. That means this virus has not entered into the tissue and the bloodstream. It has cleared directly from the lungs itself and respiratory tract itself, and not entered into any blood vessel or the tissue. Okay. So do I have time, Ravi Shankar? Because exactly 12:24, 11:24, I'm pretty sure the next speaker must be waiting. I'm ready to take any questions. Do you want me to stop here, Ravi Shankar? Ravi Shankar, can you hear me? Srinivasulu sir?
Sir, if you want, you can take 5 more minutes, sir.
5 more minutes, yeah, exactly. Very good. Okay, so let me tell you, coronavirus. Let me quote some names to this: hijacking your cellular machinery, kidnapping the cellular proteins. Okay, all these words will be very well used with virus, especially because so it is covered in spike proteins entering into the vulnerability cell. Okay, where there are ACE proteins. Okay, so we have got ACE proteins in all the intestines and all the wherever cell lines are there. We have got more in, especially in all the organs, especially liver, kidney, heart, and intestine, brain. We have everywhere the ACE2 proteins. Okay, because ACE2 protein has got some other physiological function because it controls the blood pressure. Okay, it regulates the trafficking, especially in terms of especially some signaling transaction. It plays a major role there. Okay, once it enters into the cell, releases the viral RNA, then hijacks the cellular machinery, making the viral proteins, assembling the new copies, then spreading the infection, then immune responses, and leaving the body, and finally a vaccine has to be prepared. But I'm pretty sure preparing a vaccine for COVID-19 is not that easy, it's not that simple. And I'm pretty sure that Imperial College London has prepared a vaccine, has failed in in clinical trials phase 3, and I don't know when it's going to come out. Okay, so I'm ready to take because if I go on, there are so many things because I took from evolution, I took from the cell, and I'm going much more deeper into the cell. What's happening inside the lung, and which receptor, which antibody, and which immune cell is going to attack, and how and why, and which receptor, how. So there's a lot of things. How hypoxia is occurring, how cytokine storm, and how hyperinflammation is occurring, and how the necrosis and apoptotic cell death is occurring in the lungs, and there's a lot of things to be told. And I'm pretty sure I'm given only 1 hour, and probably if time favors, probably next time I may take up the second session of COVID infection pathogens, how it enters into the lung. Look at here, how the cytokine storm occurs, the early stages, and how it becomes healthy, and why do we have a surfactant layer in the alveolus, and what happens if we do not have a sufficient surfactant layer, and why there is a lot of recruitment of immune cells in the alveoli, and why it becomes a cytokine storm. And so it's something like that. Okay, I'm trying to show you so many things. Okay, but I don't have time, so let me speak to you at a later stage. Okay, so host niches initially targeted by human invasive pathogens. Sit there. Sit. Okay, so blood. How does this virus cross the blood-brain barrier, olfactory bulb, and how do we lose the sense of smell, sense of taste, a sense of smell, sense of anything? Okay, so there are so many things, and okay, let me stop here. I'm ready to take questions. Yeah, I'm
Yes,
sure.
sir. You have so much
Yeah, there's so
information
much
and
of
so much stamina
information.
to continue this session throughout the
Yeah.
day.
So next time, a humble
Yes,
request. Next time,
yes.
whenever you give a lecture to me, you need to give me 3 hours. Okay, 1 hour is not sufficient for me.
Sure,
Okay.
sir, sure.
So yeah. So I'm pretty sure, and we may go on extended session next time. And thanks
Yes,
for giving me the
sure, sir.
opportunity. Especially many kudos to Ravi Shankar, my good friend, who has called me for this session, and for enlightening all the people. And I'm ready to take the questions now. Yeah, go ahead.
And sorry for the inconvenience caused by the sounds from
Yeah,
the unmuted, sir. Sorry.
yeah. It's okay. No problem.
If any questions, some participants posted their doubts and questions in the chat box, sir. You can go through them.
Okay. Say stop.
But even we can clear their doubts later. So our next resource person is waiting, I think.
Yeah. I'm pretty sure
And
because
thank you for such an informative session, sir. Thank you for such an informative session. Thank you for showing us the journey of COVID-2 and for giving us the details of the COVID-2 hosts like bats, pangolins, etc. Thank you very much, sir. Thank you.
yeah. Okay. Thank you, madam. Thank you, Ravi Shankar. Shall I take leave?
Yes,
Shall I take leave?
yes,
Yeah,
sir.
fine.
Okay, sir.
Thank you.
Okay, sir.
Bye-bye.
Bye-bye.
Thank you.
So, the next distinguished to honor us with his presence is Dr. C.H. Ravi Kumar, a research professor, University of Ulsan, South Korea, to present on the topic COVID-19, a pandemic. Is really remdesivir a viable treatment for COVID-19? So, a case study and synthesis protocol of remdesivir, therapeutic drug. I request Dr. P. Ramesh, Assistant Professor of Chemistry, to say some interesting facts about the resource person. Welcome, sir.
Thank you, madam. Good morning, all.
Hello, this is Dr. Ravi K. Chadrala from Korea.
Yes. Sir, mute one. Either mobile or desktop, sir. Mute one thing. Sir, we're getting a revoice. Yeah, sir. Good morning, all. Good morning, everyone. This is Dr. P. Ramesh,
Good morning,
Assistant
sir.
Professor of Chemistry.
Yes, sir.
Now, I would like to introduce Dr. C.H. Ravi Kumar. I'm elated to invite Dr. C.H. Ravi Kumar, sir, who has been serving as a research professor at University of Ulsan, South Korea. I am very much impressed after going through his research, his rich profile, which taught me what makes a researcher successful. I also understood that he has been striving hard for the betterment of humanity, which is a laudable goal. He obtained his MSc Tech and PhD in Organic Chemistry from JNTU Hyderabad. During the years 2003 to 2016, he served as a research scientist in different universities of South Korea, like Pohang University of Science and Technology, Advanced Institute of Science and Technology, and Ulsan National Institute of Science and Technology, South Korea. There, he involved in the development of organic surfaces, biomimetic actuators, nanomaterials, and graphenes. Presently, he is working as a research professor in Division of Mechanical Engineering, University of Ulsan, South Korea, and actively involved in the development of novel fluid and solid triboelectric nano generators using various novel nanomaterials. Thank you very much, sir, for accepting our invitation. Now, may I request you to share your rich experience and address the gathering. Thank you very much, sir.
Yeah. Thank you for your kind invitation and express about me. So I'm very happy to share my your knowledge since my graduation. So today I would like to express some of my feelings on COVID-19, why it is a pandemic and how to stop it. So I have to share a sparingly superficial information on pandemic information of COVID-19. Here, why COVID affects different people in different ways. So we have to find out this the serious solution from the patients. So mainly the COVID is attacking through various reasons and various ways to the human body. But after attacking the COVID, it is generally having different nausea, which is most common symptoms: fever, dry cough, and tiredness. And less common symptoms are aches, pains, sore throat, diarrhea, and conjunctivitis, headache, loss of taste or smell, or a rash of skins and other finger toes, etc. And the serious symptoms you have to find here is difficult to breathing and shortness of breath, chest pain or pressure, loss of speech or movement. These are the serious causes in the human body when the COVID is entered into the human cells. So the major affecting organ is trachea and lungs. So in the lungs trachea, the COVID is settled there and it disrupts the cells of trachea, and which is developing a number of colonies through their RNA. So introduction of coronavirus pandemic in 2019 in India, the first case entered in Kerala from Wuhan city. So the student have entered in India on January 30th of 2019, and it is attacked to several people. And as the number of confirmed COVID-19 positive cases almost 500 close at the time, and Modi has started first Janata curfew on 22nd of the same month. And at the end of the curfew, Modi started Janata curfew 2, and it is beginning COVID-19 battle against the virus. So following this, while everyone knows about, the media is expressing why COVID is entering, how to control this through lockdown, and first 21 days, and again it is going on several issues based on the controlling by the government. So if you see here, the roadmap of the coronavirus pandemic in the world and India in 2020, the background in India, especially I'm talking about Janata curfew and prohibitions of the relaxation lockdown in the phase 1 to phase 4 from 25th March to May 31st. And again, they have started unlocking in the 5 times, 1, 2, 3, 4, 5 from June 1 to October 31st. So what is the impact happening on the coronavirus pandemic? So the coronavirus is mainly focused on the economic impact, migrant workers, food supply chain, relief, impact on environment, and effectiveness. So these are the serious causes in India and drop down the economy. Almost GDP has come down to many levels. So the coronavirus cases have been reported here is almost 45 million, and the deaths are almost 1.2 million, recovered are 3.3 millions in India. And you can also observe through this web page, you can find what is the pandemic happening around the world through coronavirus. The roadmap of coronavirus transformation in USA in 2020, the United States of have been faced the coronavirus attack. It's almost 9 million, and the deaths almost reported is 0.2 million and recovered 6 million. How coronavirus vaccine works? Actually, there is one. So to stop the coronavirus, there are many medicines have been reported in the literature, but the remdesivir is the crucial drug to stop the coronavirus attack, and also after attacking the coronavirus, how to stop the mutation system in the lung and other effective organs. So you can see I'm a chemist. I can develop this remdesivir in my lab, and we have developed this remdesivir here. Just wait. So the remdesivir is the first we made from the L-alanine and phenyl phosphodichlorophosphoridate. It's the presence of triethylamine and dichloromethane. After that, it is oxidized, and we get the intermediate A and the intermediate B. And the intermediate B is again react with the intermediate C. We get a final compound. So I will tell you in the step by step how it works. Oh, the scheme was disappeared. So I'm sorry, my chemical scheme is not coming. So after formation of the remdesivir, we developed in our laboratory, and we have used for the coronavirus effect and the various applications we have done in our laboratory from Delhi. So here, it was then treated with the excess of trimethylsilane cyanide and dichloromethane at minus 178 degrees, 180 degrees Celsius. It got an intermediate of trimethylsilane triflate. That intermediate we have used for the benzyl protection. Finally, we got the liquid chromatography purification. We got the optical liquid compound as intermediate A. That is, we used as a methyl imidazole diastereomer. That stereomer is acts as a remdesivir. This remdesivir can be used as optically active drug. Remdesivir in a pure form obtained in the market. So we also developed this same compound for marketing in our laboratory from Delhi. So after using the remdesivir, many people have been recovered in 7 days. The 7 units of the remdesivir, we use 100 mg tablet or injection mode. So here, the stop pandemic of COVID-19 by filter masks, also we have studied. So the filter mask is very important here because we use a 3-layer mask, these leak-proof non-woven fabric and high-density filter layer and direct contact skin layer. So the high-density filter layer is a crucial membrane that is made from the melt-blown membrane that can be stopped less than 5 micron size water droplets from the weather. So when somebody sneezes or cough, so the whole droplets will be passed through this 3-layer membrane. But this is the very important tool for the stopping of coronavirus pandemic. So here, the 3-layer membrane is the very crucial. Here you can see the high density filter we can make with the polypropylene that is also called melt-blown membrane. It has a special kind of special kind of architecture that can stop the hydrophobic surfaces like COVID-19 having a hydrophobic surface. That surface will be stopped very effectively because it is almost less than 5 micron and more than 5 micron also we can stop here. And the inner layer, we can see the direct contact skin layer that cannot be affected with the coronavirus because it is already stopped here. And I am coming to my own research. Actually, this is my core research I wanted to explain here. This is the development of high performance ionic polymer actuators. So these development polymer actuators are very important for the mechanical and aerospace engineering. So since 5 years, we have developed several high impact ionic polymer actuators. What is the actuator? Actuator means it can be work as a robot in the space and also in the deep sea. So the basic deep sea model of robots, which are very work for the ionic polymer actuators. These are mainly depend on the different piper stacked 3D ionic network polymers and nanobiopolymer actuators based on pandemic sulfonated chitosan. As you know, chitosan is a best material from the world, from the environment. We can easily acquire from the molluscan and arthropod animals. So the arthropod animals having the chitosan surface that can be functionalized very greatly with graphene oxide, then we can develop a novel kind of actuators. These actuators can be worked as a robots in the miniature level at the space as also in the deep sea. So coming to the introduction of our research part, we have developed different actuators based on organic and inorganic and also graphene based. So the all organic NTD SPAIL actuator and nano bio actuators we also developed through biopolymers. So here you can see the research background of all organic NTD SPAIL. So the ionic actuator polymers are 2 types, mainly electronic EAP and ionic EAP. The electronic EAP is mainly 3 kinds, so dielectric and ferroelectric and electrorestrictive. And also ionic EAP is again 4 types, IPMC, ionic gel, conducting polymer, and CNT. Actually, there are disadvantages and advantages having this ionic and electronic EAPs. Mainly the advantages are mainly we can operate in room temperature conditions at long time, rapid operation time, and can hold strain under DC actuation. And in the disadvantages, they need huge amount of voltage and require compactness and between strain and stress. And the glass transition temperature also very high for the electronic EAPs. For the alternatively, we develop ionic EAPs. We can easily made in the laboratory and also through chemistry. The ionic polymers can be used for the large bending displacement, and also they have provide mostly bending actuation through longitudinal mechanism and require low voltage and bidirectional and bidirectional also. And also it generates disadvantages mainly except control properties, and ionic EAPs do not hold strain under DC voltage. Sometimes they melt also. So belonging the ionic EAPs, generally EAPs are bending very largely and also it can be take information through various the environment which is available from the uh, highest environment conditions in the low pressure and high temperatures. So here you can see the electroactive polymer, electrical stimuli responsive polymer. Actually, this subject is totally new for our environment in India, but still it is very important for the mechanical and aerospace engineering, especially radar technology. So the here EAPs can be classified into two parts: electronic and ionic type, as I explained before. Here the diamond type electronic actuator, dielectric elastomer, and electrorestrict PVDF, and liquid crystalline elastomers. So these are the important actuators we have developed in our laboratory, and we have especially made the applications of extensive, intensive progress of active functional materials have been made. Recently, many researchers are developed biomimetic artificial muscles because this cost is very cheap, and also we can easily make it from the environment, and they are eco-friendly materials which are helpful to the environment too. Because the synthetic materials are strongly disrupt the environment, because this decomposition is very long time they will take to decompose, and they will make into monomers. And here we have developed IPMC as a sandwich structure that is made up of ionic membrane and metal electrodes. These metal electrode polymers, somehow my file is corrupted. That's why some of the images are not appearing here. One moment. G. Just I want to change my file. This file is yeah. So sorry for the interruption. I changed my file now, so you can see now the real information. Okay, I just explained about the remdesivir intermediate again. Here I took the L-alanine and the phenyl phosphochloride and the TA, the DCM. We use this at 0 degrees. We got the intermediate A, and also we developed the intermediate B with the iron nose ring that is benzyl protected, and we got the intermediate B. This B and C also we made from the trizene amine pyrola compound with bromination, and we got the bromo compound. This bromo compound we developed with the N protection with the TMS chloride, and we got the TMS ring at this compound C, and these three combined, we got the remdesivir. Here, the compound B and C combined, and we got the compound D. This compound D, we have a next compound, that is compound E, using TMSCN and DCM at minus 78 degrees for 10 minutes. After the TMS OTF, we use that as a reagent, and the DCM as a solvent for the preparation of compound E. And this compound E has reacted with the compound A, and we got the compound F, and this compound F and compound A again react. We got the final drug G. This G is the remdesivir we can use for the curing of coronavirus. So how it acts, the coronavirus from the antiviral drug remdesivir. So here the SARS-CoV-2, we used to say COVID-19 when it penetrates into the human body. So it replicates and it forms a RNA replication very fast, and virus is making into colonies in the body. So these colonies have been stopped by attacking of this remdesivir. It is having here, you can see the RDRP. This RDRP coronavirus, the RNA will be hacked by the remdesivir, and this mutation will be stopped, and it will be killed by the virus RNA. So the RNA was hijacked by the remdesivir. That is the reason this is working very well for the compared to the other SARS drugs and antiviral drugs in the market. So remdesivir, we are also developing in our laboratory in the New Delhi as our collaborative laboratory. You can see here the remdesivir potential repurposed drug candidate for COVID-19. So here the RNA channels or RNA virus of the RNA is almost growing a lot after incorporating the GS441524 drug as a remdesivir analog. It will be reacted strongly with the RNA of the uh, coronavirus, and it will be stopped here RDRP, and it is almost RNA is stopped growing, and remdesivir is working well as GS441524. So this is the real information I would like to share previously, but unfortunately the file was changed. And the three-ply mask also very important. People are just ignoring about the mask. Actually, mask is the primary tool to stop virus. So please don't ignore the mask, because you have to wear the mask, because the mask is having three important layers, which are mainly inner layer, outer layer, and also middle layer. So the middle layer is the key layer. This is made with the polypropylene melt-blown membrane. This membrane is very crucial for stopping the virus, because the virus having the hydrophobic surface with the water drops. So these water drops will be accumulated on the surface of first layer, and some of them are going to the second layer, which are arrested here, and they will be killed through the hydrophobic surface interaction. They will be stopped, and the RNA surface also ruptured here, and it will be stopped here and killed by them. And also the accumulated virus is simply eliminated, because you can throw after 24 hours of this membrane, you can use a fresh membrane. So this is the fundamental tool. Everyone wanted to use it, but many people are ignoring in India and everywhere. So we are developing the three-layer membrane also in our laboratory through our surface coatings. And I already explained here, the aerosols and water droplets generally pass around 10 micron, the size of the water droplet, that will be passed through first layer, and it is arrested here in the second layer. This is a melt-blown membrane, as I said. This is made of polypropylene surface. So this polypropylene surface is already having hydrophobic surface, hydrophobic interactions. Many of the virus droplets are arrested here, and they will be stopped continuously, and they will permanently stop at the second layer. And the outer layer of inner layer, it is showing direct contact with the skin. This is very smooth layer, and also it is made with nanowoven fabric. So these three layers are very important for the COVID-19 before using or before entering into the virus into our body or of the drug. And now I wanted to stop about the coronavirus, because coronavirus I am not focusing very much due to request of my friend. I just giving the superficial information about the remdesivir and three-ply mask and five-ply mask. The five-ply mask is made of different nanowoven membranes with the 80 gram of the polypropylene, that is only used for the medical doctors. That is the use for the medical doctors, because they wanted to use for more than 1 week the same film for the not entering the coronavirus. And I'm continuing my original research here. So now you can see here the traditional biomimetics made from the electroactive polymers, which are we can replica of the natural birds or natural animals in our laboratory. You can see the web of the duck here, and the artificial muzzle we developed here. You can easily observe with my mark. And also we developed a special kind of wing for the rocket that can be moved into the space. That is a replica of the bird wing. And also you can see the nectar. This is also for we can see, and also we developed a simple nectar material through our laboratory, and also paper type electroactive polymers also we have developed. And also we developed a tympanum layer. This layer is very important for the audibility in the human ear, internal ear. That also we developed in our laboratory for developing the conicalia material, and also with the graphene mixed with electroactive polymer. So this material are very important. We have taken for the patent from the US, and also we have the artificial modules. Those modules we developed in our laboratory. These laboratory can act as a high stretch and strains. We can develop here. These stress strain materials also we have developed in our laboratory. These are biomimetic actuators. These are also called artificial modules. And these artificial modules are mainly made of electroactive polymers. These electroactive polymers, as I explained earlier, electroactive, electrorestrictive, electronic, and ferroelectric. So the ferroelectric materials are made up of iron nanoparticles. These iron nanoparticles are not longerly stay because they will be oxidized by the environment. So that's why it is not working very well for the electroactive polymers. So to overcome these strong drawbacks in the science and technology, we are developed a novel materials which are ionic EAPs, because these materials are made up of a network of ions. Those ions are strongly binding with the polymer. This polymer is very good, both water and oil. So these oil and water materials are strongly bind with the ions. They can be spread very easily within the whole polymer, and it acts as a ionic polymeric material. This material can be moved by the electrons, and when you charge applying on the surface, this will be move off like a actuator. And also there are other three materials we have developed, that is ionic gel, conducting polymer, and carbon nanotube materials. So as I explained earlier about advantages and disadvantages of ionic actuators and what is the reason we developed here, we already explained here, that is IPMC is very important material for our laboratory and in the science and technology, especially in the space and aerospace materials. And you can see here some of the images and movies you can find we developed, and also some of them have been developed in the various universities in the world, like MIT, Michigan State Institute of Technology have developed diamond type of electric actuator. It can be moved from longitudinally from top and down when you apply the voltage up to 5 volts. And there is a dielectric elastomer actuator. It can be a hexapod robot. It can walk in the space through our ionic electroactive polymers. And also there is electrorestrictive polymer. It is made with the PVDF material as a electroactive material. It can be worked as both hydrophobic and as well as hydrophilic material. This can be strongly worked here, and it will be moved from top and down. And there is another liquid crystalline elastomer. This also we have developed earlier in our laboratory. And there is EFP actuator we have developed from Inha University, and conducting polymer actuator. These are the state-of-the-art materials now in Korea and everywhere, because this is the very important material we can keep in the deep sea to detect the sonar signals from the sea, and also we can also detect from the earthquake waves through our system. And the final material we developed, that is IPMC actuator. This is ionic polymer metal active composite. So this material is made up of polymer, but this polymer is deposited by the metal coating, because the metal coating is very crucial to pass the charge. So to charging the material, we have to develop a sandwich material. That sandwich material will be coated by the either gold or silver or platinum metal we use for the charging of the voltage. And there are several deposited and reported materials in the world, and from various universities can see the Braille cell, photo detector, and a Rafish robot and a speaker. We can easily audibility at the nanoscale level voice, and also energy harvesting when you walk on the surface through our shoe. It is modified in the surface of the bottom of the layer. We deposited the sole of the shoe, and we make a grids like this. When it walks, automatically the charge will be generated through turboelectric nano generators. And also we developed a jellyfish robot, and the solar cells also we have developed. This is transparent solar cell, which is very important material for the capturing of solar energy through our system. And also dust wiper. This is we can also develop in the where the human being cannot go inside. There our robot will go inside, and it will clean the surfaces like inside the iPhones or inside the uh, TV kits or computer kits like that. Our system will go and clean the surface. And the flexible display monitors also we have developed, and actuators and sensors, and also stents. You know, stents are very important in the medical technology now. The stents are very useful through carbon nanotechnology. So we developed a special kind of stent that can be used for the heart attack, and also cleaning of the arteries and the blood vessels in the human body. So we have developed a simple material which is using for the Nafion. because naphthalene is a commercially available nanomaterial supplying from the Dow company from Germany. So this naphthalene is the key intermediate, the perfluorosulfonate naphthalene, which containing sulfonic acid as a tiling error. So this tile material can be used as SO3H. The SO3H can be replaced by any metal. Generally, we use lithium because lithium is a very good metal, which is the very close to hydrogen atomic number. That's why we use the lithium metal. So the lithium ion can be incorporated on the surface of this sulfone naphthalene that can be deposited here. You can see the semi-mesh. The upper part, you can see the surface of the lithium ion. When the actuation starts, the ions inside the ions of the actuator, they can be moved when you apply the voltage. The voltage, when you apply, automatically the ions will be stretched and released. When it is stretching, the ions will be oppositely move into the opposite electrode. Like if it is a negative electrode, the positive ions will be moved towards the negative electrode. And when the charge is increasing, automatically the positive electrode attract the negative ions in the within the network. So these ionic bondings are very flexible. They can be easily mobile, and they can be strongly bind with the whole host polymer. And the lithium ions or any ionic bond can be easily move from one end to the other end by the charge applying through. When the charge is increasing by voltage, automatically the movement is also increasing through this network. So you can see here the animation. So here the 2 volt DC voltage, the movement is slowly moving at the 0.2 hertz. When the voltage is increasing, the movement is also gradually increases up to from 0.2 hertz to the 16 hertz. So there is large deformation also you can find. This is the robot we developed. This is very crucial for the aerospace technology and rocket technology for incorporating of our materials and mechanical engineering applications. And this is the fundamental uh, polyelectrolyte system. You can see the network. This network is hosting with the different ions. These ions are strongly binding with the sulfonic acid. This sulfonic acid is covered with the electrode, as I said earlier, about platinum and gold or silver we use as the material of electrode. These electrodes are both sides. They can be connected to the uh, voltage. They can be moved when the voltage is increased, as I explained here, as IPMC actuator has many advantages through large bending deformation under low voltage. In generally, people using 150 volts to move this kind of actuator, but we use only just 2 volts to 5 volts. This is the key achievement we got from our laboratory, and we published various papers in advanced materials and nano energy earlier, and also we are publishing different MRS bulletin. Earlier we published in 2008. And there is a construction of conventional IPMC actuator. There are the fundamental important requirements to construct this kind of polymers. Mainly block copolymer is very important, and blocking force is also very important. When the actuator is moving, sometimes the actuator will be break because of the stretching frequency is very high. Automatically, the stress and strains is also increases. The polymer will be break due to the voltage. To overcome that, we have developed a simple material, that is we take from the chitosan. The chitosan is a very important material from the nature, which is abundantly available from the molluscan and arthropodon family in the zoology. So here you can see synthetic polymer also we have developed for the making of different actuator. So here we developed a all organic NTDA SPI ionic liquid actuator. Actually, ionic liquids are a key material for the large ionic density, and they can be strongly move in the high voltages and room temperature. They will be act as a liquids. These hydrophilic and hydrophobic moieties combined together in the ionic liquids, they can be used through penetration of hydrophilic channels and hydrophobic channels of our polymer. This is our polymer, sulfonated polyamide. This I have been developed in our laboratory, and we make into a polymeric solution. This solution as stacked here, you can see here the semi-maze. The semi-maze having the 3D zigzag network. This network having the ions attaching with the sulfonic acid. This is also, we can say, pipette stacked LBL structure for the NTDA SPI SO3Li EMI TF2N membrane. This membrane is very crucial for the making of different actuators. Also, you can see the self-assembly by pipette stack 3D network membrane. There are three key features. There are four features we have developed here: pipette stack, layer by layer structure, hydrophilic, hydrophobic, ionic interactions. These are the key important factors for us. And the ionic hopping mechanism, because somebody has to push from the backside, then only the ion will be moved. That's why we use ionic liquid for the pushing. That's why this ionic hopping mechanism is very important for here. And also 3D zigzag cross-linked nano channels, because the nano channels are, even though we developed layer by layer, but they have interconnecting each other. These interconnecting also having some positive and negative ions, which are very strongly binding each other. And the strongness is almost having the uh, stress strain should be compatible, that can be helpful to break, helpful to stop breaking of the actuator. So here you can see the zigzag network by CC bond rotation and also forming of the pipe stack by layer on layer network. So these materials are very important and they are very stable. Even at 80 degrees also it is very strong enough. And even we checked at 150 degrees, it is worked as the same as a normal room temperature. And we developed this actuator through chemical modification. We also developed non-conducting material, that is, non-metallic material, that is conducting polymer, P.PSS we use. This P.PSS is also a conducting polymer. This is compatible with all kinds of uh, organic liquids, especially we use DMSO, DMF, and N-methyl pyrrolidine. And this is the synthetic scheme we developed for the making of this actuator. You can chemistry people can understand. You can see the BDSA. This is having two amines at the both sides. They can be protected. The sulfonic acid is protected with a triethylamine in the presence of metacresol at room temperature 4 hours, and we temporarily protecting the triethylamine salt. We got the BDSA TE salt. This we use as a monomer, and the other two monomers we use NTDA 2 moles and ODA is 1 mole. And this block of polymerization, we made it in same metacresol solution at 180 degrees and benzoic acid as a uh, actuator, activator for this reaction. And after 24 hours, we got the linear polymer of PDA and NTD as BDSA salt. This salt will be hydrolyzed in 1M HCl, and we got free sulfonic acid at the base structure. And the sulfonic acid can be again metallized by the lithium chloride. We got the methylated or lithiated compound. This we use for the sulfonated actuator for the polymerization and also for the development of metals and non-metal conducting polymers on surface. So here the preparation of different actuators we have developed with the 3D network, and also I explained earlier how we developed this SPI solution and ionic liquid. We combine together for 24 hours or interact each other. The ions will be penetrate and they can be deposited, and we make into a membrane. That membrane we cut through the Dr. blade, and we developed a special layer on top of the uh, conducting polymer. This special layer is a P.PSS. This is a conducting layer. That conducting layer will be very strongly glued on the surface of the base network. So this is the actuator. This actuator morphology it looks like, and the chemical structure should be like this. And you can also observe the ionic liquid on top of the surface of conducting polymer. So when we attach the electrode to both sides, automatically these electrodes can be moved when voltage is increases from 1 to 5 volts. And this is the 3D network model we have been developed earlier. And this is the morphology of the different actuators at TES stage and the sulfonic acid stage. After removing the TEA, there is a holes we've observed on the surface. It means the TEA molecular weight is almost 107. After removing that, the surface is having some pores and ridges. These pores are because of the removal of TEA salt, and we deposited again lithium ion. So when the lithium ion is deposited, you can see the circle. There is a metal on the surface, and also the pores have been filled through the metal ion clusters. These clusters you can easily observe in the cross-sectional view, the figure F and figure E. You can see the ionic liquid deposition. The ionic liquid deposition is brightly surfacely incorporated, and they can be layer on layer deposition. They can be interacted very easily at the surface of ionic metals, which are mainly sulfonic acid and lithium ion. And we also determined with FTIR and the improvement of actuator, from chemistry part of view. And we check the tensile modulus. This is a tensile modulus is very important for our actuator because the stress strain is very important. We developed this material almost 1.5 to gigapascals. We developed from zero, and also after incorporation of ionic liquid, the stress strain is increased almost 1.5 to 1.7 to gigapascal. It means that the ionic liquid is acting as a surface plasticizer. This surface plasticizer will be useful for the movement of ions very strongly. So you can easily observe this image A, the stress and strain, though you can see the NTDA space O3HTA is a dry mode, almost it is 9, but when we incorporate all the materials, we got almost 34 strain percent, and strain stress is almost 54. The gigapascal tensile modulus is almost enhanced from 0 to 1.725 gigapascal. This is the great achievement we got from our laboratory. And also we check the morphology and X-ray diffraction of the crystallinity of the material. The material is gradually crystalline because of the ionic liquids are deposited very strongly on the surface of base polymer. So the crystallinity has been increased from sulfonic acid to sulfonic acid Li, EMI, MTF2N. You can observe in the figure B. And this is the key application of our research. This is the movement of tip. Tip means the actuator. So the actuator generally moves just 0.1 millimeter, but after a deposition of our polymer and we developed in conducting polymer through P.PSS, there are three sine waves you can observe here at 2.5 volts, 0.1 hertz oscillation. So the movement of the tip is gradually increased from black line to the red line, the black dotted line, blue dotted line, and the red strong line. So the red strong line is showing the tip movement from 0 to almost plus 3 to plus minus 3 tip displacement of millimeters. This is the key important, but this is the literature we developed very first time, and this paper we have published in Advanced Functional Material recently. And also we developed this material with different oscillating external frequency. We observed the driving voltage when we increase from 0.5 to 2 volts. So the maximum 2 volts we applied to at different hertz, 0.1, 0.05, and 0.01. So the 0.1 is the highest oscillating voltage, oscillating measurement we took as a control method. But even at lower applied hertz, and we get the almost greater tip displacement we observed with 0.5 volts at 0.1 hertz of the tip displacement after an actuation test for 1 hour. Generally, the actuation is happening for the 10 minutes, but we tested for 1 hour. This is almost very long period. It means that the actuator is, the shelf life is almost 1 hour, means it is definitely comparatively very advantageous with existing actuators in the literature. And this is the voltage displacement, 0 volts, it is almost in the straightening. When we use a negative voltage, almost it is moved towards the left side, and it is a positive voltage, it is into the right side. It means that 0.1 hertz frequency, the excitation frequency, when we applied, the tip displacement is very high, even at low frequency. But in the commercial materials made with Naphthalene, because Naphthalene is almost generally having oxidation property, it is oxidizing and the polymer is almost breaking during the actuation process. This is the drawback we observed from the literature. For that, we have developed several actuators. We published various papers in nano energy and also advanced functional materials since 5 years. Also, we are developing stents for the arteries and the heart beating to enhance the heartbeat and to regulate the heart beating. Stents also we developed in our laboratory. So thank you for your kind understanding about our research area. So this is a very state-of-art research facility we have in our laboratory, and COVID is my part of research because I am the collaborative company CEO. So my company, collaborative company, is making of remdesivir. So we are marketing in India and abroad. through our company in Delhi, and Noida is the laboratory we are existing, having the company core facility. So we are also supplying remdesivir for the marketing through our company that is called VRND Nanobioorganics. Thank you for your attention. And if you have any strong questions, I am ready to. Hello.
Yes, sir. Yes, sir. Any questions from the participants? Or you can post your doubts in the chat box. So now I express my sense of gratitude to you for sharing your valuable thoughts and useful observations on the antiviral drug remdesivir. Thank you, sir, for sharing your knowledge on electroactive polymers through the animated slides. They were very interesting. And thank you very much, sir.
Welcome. So thank you for giving me a great opportunity and interact with you, intensified and knowledgeable people in India, and especially in our states, Telugu states. So I am really grateful to invite me. So thank you for organizers and also Dr. Ravi Shankar, who invested a lot of time to organize this webinar.
Okay. Sir, one participant is ready to ask a question.
Sure.
Sir, yes. Sir, ask.
So due to time constraint, I have not given many information.
Next time we'll give you more time,
Yeah,
sir.
sure.
Thank you, sir. Are you asking the question? No. So I will continue our session.
Ma'am,
Next,
one question.
we look forward for. Yes. Okay. Okay. Continue.
For me, it is one
Continue.
question,
Please
please.
continue. Yes. Okay. Okay.
Sir, this is really fantastic information you have given to us. Really, we are very happy to say this, sir. But my straight question is, actually, when we are using masks, actually, I have raised that question in previous session also. So 3 layers, you have talked about the 3 layers. In that, the size of pores in each layer, we have to compare with the size of virus, the size of the virus. So at that time, how they are going to prepare to resist the virus with the size of pores in the layers?
Actually, your intelligent question is very nice. It is also key question for many people, even for the literate and unliterate people. Mainly, the layers, we made 3 different layers. I have very good experience on the surface coatings. That's why I mentioned about the 3 layers. So the middle layer is the key layer for stopping the virus because it is made with melt-blown membrane.
Sure.
So the melt-blown polypropylene membrane having 2 gauges, 20 gram gauge, 80 gram gauge. Okay, 20 gram gauge, the pore size is bigger. 80 gram size, the pore size is small. So that's why the doctors are using 80 gram melt-blown membrane. That is 5-layer membrane. You know, that is 3M1860.
Sure.
So the 3M membranes, I mean 3M company from USA, they are making 1860. They can stop the, the membrane can stop the virus even at 100 nanometer width. But generally the virus size is 100 nanometer. But the droplet size is more than 5 micron. So the 3 layers membrane is only stop for the droplet from the other person spreading from his sneezing or from his cough. So the cough having the droplets, this is containing our water droplets as well as the virus colonies. The colony can be interact each other and they make as a colony. The colony should be having less than 1 micron to 5 micron along with the water droplet. That's why they will arrest in the middle layer.
Sure. Thank you for a good answer, sir. And the little question also is there with concern with this. Actually, our skin also is nothing but a porous layer. We can treat that also. So the bore of the that sweat glands, such are also bigger than the size of the virus. So we are talking ever and ever. The three gateways are there: nose and mouth and eyes. Through them only virus enters. No, it is technically I'm feeling a little bit hard to receive that. Why? Because the skin also has
Yeah,
and when
I
virus
think,
falls on our skin, also it goes to inside the body. So I want to
yeah, Mr.
understand.
Let me explain about that.
Please, sir.
From the skin, you can get the sweat. Sweat is
Sure.
hydrophilic.
Sure.
So the sweat is having ions, sodium chloride ion.
Sure.
Okay.
Sure.
So the sodium chloride ion can be stop any hydrophobic material.
Sure.
So
Sure.
coronavirus surface is having hydrophobic chain.
Sure.
So the hydrophobic chain will not enter through our skin.
Okay. Okay, sir. Sir. Sir.
Okay. Although your skin is porous,
Sir.
but it releases the hydrophilic liquid like sodium chloride.
Sir. Sir.
That is around 1% or less than 1% of the volume. If you compare with the weight percent.
Sir,
So
good
that can be destroy the coronavirus from the skin. That will not enter through skin.
Sir, good answer. Thank you, sir. Thank you, sir. I'm satisfied.
So our nose and mouth as well as eyes are very good windows to pass through the coronavirus.
Sir, sir. Thank you, sir. Thank you very much for your valuable.
So we are also making masks in our laboratory.
Sir.
Yeah. So very soon we are going to introduce in India.
Thank you, sir. Thank you.
Thank you for giving me a great opportunity to interact with you people.
Thank you for the nice interaction, sir.
Yeah,
Thank
somehow,
you very much.
somewhere we got wrong selection of our file. That's why I disturbed, but it is okay.
But they were interesting. The slides were very interesting, you showed. Right? Thank you.
Thank you.
Sir. So next, we look forward for the presence of Dr. N.V. Raman, a research scientist in drug discovery, Eurofins Limited, the United Kingdom, on the topic development of inhaled target drugs and potential treatment for COVID-19. I request my friend Dr. C.H. Chaitanya, Assistant Professor of Botany, to give a short bio of the research person. Welcome, ma'am.
Thank you, ma'am. As we know, human beings' search for panacea is as old as pain, which means since the inception of human race. Identifying medicines for diseases or disorders is a painful job with multi-layered and complex issues. So I think at least with the 3 extraordinary lectures what we had from the morning, all of us might have got an overview of this hectic work. So no doubt that drug design and its administration is not as easy as it appears to be. So now I'm excited and delighted to introduce our next speaker, Dr. N.V. Raman, one of the most renowned scientists in drug discovery. He received his PhD in medical chemistry from Milton Keynes University, UK. He pursued his postdoctoral research in bioorganic and medicinal chemistry at Milton Keynes University, UK. Then he joined Argentina Drug Discovery Limited in 2006 and rendered his services for 10 years. Later, he moved to Charles River Early Discovery Services, UK, and he is currently working in Eurofins Limited, UK. His area of expertise include synthesis of biologically active molecules related to various diseases such as cancer, respiratory diseases, rheumatoid arthritis, etc. I feel that Dr. N.V. Raman's parents must have named him after a great scientist, Sri C.V. Raman. True to his name, Dr. N.V. Raman, sir, achieved many laurels in his area of research. Thank you so much, sir, for accepting our invitation despite of your busy schedule. So without any delay, I now welcome Dr. N.V. Raman sir to share and enlighten us with his expertise. Thank you, sir.
Good afternoon, everyone. Can you hear me properly?
Yes, sir. Yes, sir. Very good afternoon, sir.
Hi, good afternoon. Yeah, how are you? Okay.
Nice, sir.
Yes, sir.
Good,
Very
good.
good,
Yeah.
sir.
Thank you very much for inviting me to this webinar. And I would like to, I really appreciate all the organizers, really well organized. And today I'm going to talk about development of inhaled target drugs, which are the potential treatment for COVID-19. So I work as a senior research scientist and in drug discovery and medicinal chemistry. So I probably some of the slides may repeat as the previous speakers. So I didn't go through all the slides of the previous speakers. If they repeat, sorry about that. So today my contents of my talk are a little bit of background about COVID and current market drugs for COVID, and a bit of flavor about vaccine and clinical trials, and I'll explain something about my current project, which target for respiratory diseases, and conclusion of my project. And if time permits, I'll give some health and safety tips. I'm sure everyone know about those. I'm sure, but if you see the pictorial presentations, then you will easily remember those. So moving on to next slide. So every day when you wake up in the morning, the world is talking about the coronavirus and how it spreads. So just as introduction, so COVID is a coronavirus disease 19. It starts in 2019 October roughly in Wuhan. So a number of countries affected and a number of people affected, and with the symptoms like a cough, cold, flu, and spreads through droplets. So as previous speakers mentioned, and main thing is trick is social distancing here. So if there is no social distancing, it can spread easily. Moving on to next slide. Right. So basically, so when you're traveling or when you're working in somewhere, like touching the contaminated objects, so that will directly if you touch something contaminated and touch with the hands with a mouse, nose and ears or eyes, whatever it can go through. And WHO declares this is a pandemic situation in March. So a large number of countries affected. We are in a very unprecedented situation at the moment. So this big picture shows how virus go through the through your mouth or nose into the lungs. So target of this virus is lungs, basically the main part of the body. So moving on to next slide. So the symptoms, I'm sure. So the major symptoms developed initially when coronavirus started. These symptoms were not there, but later on observed like sudden loss of smell and taste, shortness of breath and all those. Then other symptoms like the rashes in the skin and all those. So some people might have symptoms, some people might not have symptoms. So it can be severe, it can be starts in a week or 2 weeks. So next slide. So why it is so deadly? Because coronavirus target is it can easily go through the lungs and airways, so which will attack your immune system. Because this pandemic was observed in 1918, which was Spanish flu. So almost death rate was estimated death rate was 50 million people worldwide. So basically the reason for stick to the lungs is so coronavirus contains 70% of fat. So it can easily stick to the lungs. Once you're infected, it won't come out easily unless you are healthy lungs. If you do like the breathing exercise and all those, unless you have very strong immune systems, especially for the old age people, this coronavirus, once it sticks to the lungs, it will damage the lung cells. So the one key thing here is Normally, under normal conditions, our body, when something like another, if you enters into your body like bacteria or virus, the body cells start releasing chemicals which are called as interferons. So once they are being hijacked by any virus, your body will give warning signal to the rest of the body. So then your immune system will alert. So then that can kill the virus. But in case of coronavirus, it can block that warning the warning system by the body. So that's why you don't see any symptoms that easily. So it can rapidly grow after like another week or 2 weeks. Then once it go to the lungs, it will simply kill the lung cells. So especially depends upon the age as well. When you are obese, like when you are overweight and all those, so that will affect a lot. Because in the old age people, like they'll produce a lot of less T cells, which are quite important in the body. I don't know what happened here. Sorry. So because the obese people, they'll have excess of fat in the organs like liver, kidneys, and all those everywhere in the body. So they will have less metabolic activity, and there are more chances to get the damage by the damage those organs by coronavirus. I'm sorry, something happened on the screen. Is the yes, yeah. So okay, so what is it? What are the treatments here for the coronavirus, and is there any diagnosis and treatment? So far, you can see in the worldwide what's happening. There is no specific treatment for coronavirus. So basically, prevention is better than cure for in this case, especially. COVID symptoms very similar to the other illnesses like the flu, cough, cold, and all those. So only test we have is so far is a swab test, and once it is positive, you need to take care. You need to look after yourself. You need to be preventive. So if you have the mild symptoms, stay home with home quarantine, uh, with usual medicines like the hydroxychloroquine, remdesivir, and all those. So usual precautions like face mask, social distancing, hand washing, and alcohol-based sanitizers you need to use, and you need to seek medical advice on all those. So basically, the simple tips make you be careful about the coronavirus. And moving on to the next slide. So what are the current drugs in the market? Okay, as the previous speaker, Ravi Kumar mentioned, remdesivir as a popular drug, and hydroxychloroquine and favipiravir. All those drugs showed very good antiviral activity before. So and proof of efficacy against the COVID-19 as well. So all these drugs, some countries licensed to treat, some countries didn't had the license to treat these. So this favipiravir, for example, is still in phase 3 clinical study. So moving on to next slide. So some of the drugs like umifenavir and ribavirin and niclosamide, all these previously showed antiviral activity against different viruses, and also tested against the COVID-19 as well. So, but not all the countries using for all these medicines. All right. So moving on to the next slide. So the three other popular medicines, oseltamivir and dexamethasone, azithromycin. So if you take the dexamethasone, this has been used for the respiratory drug when people are severely ill, and this is only suitable for when people are on the ventilator in the hospital. So old age people. So which is approved by NHS UK government, because lots of deaths in UK, about 65,000 deaths. And azithromycin, and this has to be used along with hydroxychloroquine, which is it's an antibiotic, so antiviral, and so basically the antiviral against the number of viruses, and also tested against COVID-19, which works initially really well, but are not 100% cure for the coronavirus. So moving on to the next slide. So another form of treatment is vaccine. So world is talking about the vaccine every day now. So we are thinking when the vaccine, why it is vaccine, why the vaccine is late, why companies are not bringing the vaccine out faster. So then you will see in the next slides what's happening with the vaccine. So just as a basic introduction, what is vaccination? So vaccination is the safest way to protect you against another diseases. So once you have been vaccinated, your immune system will develop, that will produce antibodies, and that will, sorry, just 1 second, this video is blocking me here. So once you've been vaccinated, your immune system will alert and the fight with the disease. So how the company is making vaccine now? So the first, what they will do is, in the vaccine, the main ingredients are, what they'll do is, whatever, for example, you are targeting coronavirus to kill the coronavirus. What they will do is they'll take the coronavirus, they use the killed coronavirus or greatly weakened virus or broken parts of the virus, which is not harmful to the body. So they'll make that's the main ingredient in the vaccine, and other ingredients are water, stabilizers, and other preservatives. So once they make the vaccine, this is not harmful to the body at all. So moving on to next slide. So under normal conditions, what will happen is our immune system always fight with the diseases, like whatever, like malaria, dengue, and other diseases. So when virus or bacteria enters into your body, if you take a medicine, like your immune system will recognize and recognize this some harmful thing enter into your body, so that will destroy it. Your immune system will destroy the virus or bacteria. So these, what immune system, what will do is that will produce antibodies. So antibodies, like acts as soldiers, all those. So once the infection is gone, for example, if you got the disease again, the same disease again, your immune system is a bit of clever ways, so it will respond. Like you got to memorize the disease again and rapidly, quickly destroy that virus again, and you won't get the disease again. But move on to next slide. Sometimes your immune system depends upon your body, person to person is different. Your immune system may not always fight against the harmful bacteria or viruses. The best example we can see is COVID-19. Excuse me. So we need the vaccination for the new viruses. So once, what they'll do is, as I mentioned about the making of vaccine, it contains non-harmful virus, coronavirus in that vaccine. Once you got vaccinated, your immune system will alert and produce antibodies, and then attack that the non-harmful virus. So basically, your body is ready now. So like with antibodies. So if coronavirus affect you, so that antibodies, your immune system will alert, like you got a memory that you got same disease. So if antibodies, so same foreign object enter into your body, then that will antibodies attack the coronavirus and kill it. So moving on to the next slide, you can see the right side in the pictorial representation in slide 14, how they make the vaccine, how the antibody is produced. So we are very eagerly waiting for the vaccine. So what are the stages of the vaccine development process? So there are lots of challenges. So we are expecting in just within 6 months, but you can see in this in the next couple of slides what's happening. So first of all, FDA, once you develop the vaccine, FDA approves vaccines for the in vitro and animal testing and clinical trials. So what these researchers do is they use the cell cell cultures, tissue cultures, and the other animals like mice, monkeys, before the clinical trials. So they have to confirm that vaccine is safe before come to the clinical trials. And then they have to make the application to FDA to test on the humans. So because we are in the pandemic situation, currently we need the vaccine very rapidly. So all these companies trying to develop the vaccine, and also FDA is supporting them to approve as much as possible. So you can see the picture here. So need to discover the vaccine, and they do develop the process development, and then clinical development, phase 1, 2, 3, and then assay development, whether that is pure and stable and it is working or not. So these are the stages. So moving on to the next slide. So the challenges are, for example, sometimes when you're making virus vaccine, the vaccine contains, as I said, like a virus or bacteria that might create complex problems. And it should be cheaper to supply for millions of people, economically viable. And it should be pure and stable. And storage might be a problem, like the storage and the refrigeration. Some vaccines need to be stored at certain temperature. And when they're making vaccine, mutations might occur in the vaccine, causes a lot of problems in developing a vaccine. So need to change the vaccine constantly in that case. So you can see the the picture in the bottom here. So average phase length, at least a minimum 2 to 5 years. Actually, the vaccine, if the vaccine properly need to be worked. So once they develop the vaccine, they do the cell culture, animal studies, and all those. Then they need to pass through. Once it passed through phase 1, phase 2, phase 3, phase 4. So next slide, I'm going to explain about clinical trials, bit of background. What are the clinical trials? So everybody, we are reading in the newspapers, some drug, AstraZeneca, Oxford, they're all doing clinical trials. So what are the clinical trials? The clinical trials are, basically the simplest way to say is how effective the drug is, how how safe the drug is, how effective, how it is working effectively in the people. So that's the best way to find whether the drug is working or not. Those are called as clinical trials. So clinical trials are two types. One is observational studies. So what they do is they gather group of volunteers, and they gain a lot of information by asking about health conditions, medical examination tests, lots of survey they will do. And based on this survey and data they collected, so they might be helpful in identify the new possibilities in clinical trials. So moving on to next slide. So the second category is clinical studies. So this is the real testing. So basically this is these studies involving people directly, testing medicine directly onto the people. So it involves 4 phases. So here it will advance to 4 phases. Once if it passes first 3 phases, US FDA approves the drug as a clinical use and continues to monitor the drug in the 4th phase. But before these clinical trials, all the researchers, scientists, what they will do in the laboratory, they'll do the animal studies to make there's no side effects of the drug and it is effective and safety. So they'll make sure that then they'll apply to the FDA. So then FDA will approve for the clinical trials. So these clinical trials, moving on to next slide, slide 19. Moving on to the next slide, the phase 1, the vaccine or medicine, new drug, will give it to the 20 to 80 people first. They'll monitor, is there any side effects? It is safe enough? And is it the correct drug dosage? Once it passes phase 1, it'll go to phase 2. They'll take another up to 300 people, and most people will be ill, the disease they are trying to treat, and they will check, they'll monitor whether the drug is safety and effective, it is working or okay or not. And this phase can be last long as several years, maybe 2-3 years here, phase 2. And phase 3, once it needs to be passed phase 1 and 2, and once it passed to phase 3, and they will use 3,000 people to test the medicine. So if the results are positive at this stage, FDA will approve the drug, and then go to phase 4. So safety, what they will do is, and the phase 4, the FDA approves, they'll use thousands of people to test the medicine in phase 4. And safety and side effects they will monitor, and how the drug is affecting, how it is working, safe or not. Finally, what they will give, they will give the marketing license and available on prescription in the doctors. So you can see the pictorial representation here in the bottom. So first they will do the preclinical studies here in the laboratory, then goes to phase 1, phase 2, phase 3, and phase 4. So moving on to next slide. All right, what I am doing now. Sorry. So what is the link between my project and COVID-19, like another respiratory disease, basically? So my current my project was completely project disease. My current project is inhaled target drugs. So the specific goal of the project is identification of selective kinase inhibitors. So kinase is an enzyme, the number of kinases in our body. So we're targeting, like, the specific kinase inhibitors, kinase inhibitor drugs. So this project is in partnership with the Genentech USA. So this is integrated drug discovery with chemistry and biology. And I was working as a senior synthetic organic chemist and medicinal chemist. Moving on to next slide. So what are the current kinase inhibitors in the market? So lots of companies were trying to find the drugs, discovery of drugs in the market, like in AstraZeneca, like Pfizer, GlaxoSmithKline. They're all working on kinase inhibitors, a lot of targeting the respiratory diseases, because especially in these countries, COPD, asthma, the number of cases, like in the highest number of cases these people will have, because lots of smoking here. So competitor companies, what they're working is they're working lots of drugs, but the thing is, the major drawback they have is they're having lots of side effects, like diarrhea, headache, high BP, and fever, nausea, vomiting, etc. So because all these, because of side effects, they are failing in clinical trials. So the goal of my project is the discovery of selective kinase inhibitors. So moving on to next slide. Right. So just a few example. What are the current drugs in the market? So salbutamol, Ventolin. I'm sure many people know about this. It's a short-acting inhaler drug. Whenever you go to the doctor, if you got the breathing problems, so first drug to prescribe is a salbutamol with a small dosage. What will do? It will do is it will open up the airways, like another. So when they'll give the inhaler, when you put in the mouth, inhale, so that will open up the airways. So which you always need to carry, whoever got the asthma. So they need to carry in their pocket all the way. So salmeterol. So this is used for COPD disease and asthma symptoms as well. The same thing. And formoterol and vilanterol. So all these are bronchodilator drugs, and to open up the air passages in the lungs to make easier to breathe. So these drugs, what happened is when the GP doctor prescribe these drugs, you need to take the inhaler with you always. Whenever it is necessary, you need to inhale, or like maybe every 2 hours, maybe every 5 hours, 4 hours, 1 hour, we don't know, but depends upon the condition of the patient. But what we are targeting is, moving on to the next slide. So what we design the target of the what we are targeting is, for example, if we make a drug, if you inhale just once in a day, like a morning, just one puff, just leave it at home, and it should work as a long-acting bronchodilator. That's our target. So we should not the use the many times in the day. So that's one puff is our target, but the thing is we don't know is works or not. But we made this, and these are in the clinical trials at the moment, advanced studies at the moment. So what we did here, so we did it from our previous projects here, and we did another X-ray co-crystal and docking studies on these molecules, and which suggests like this right side tricyclic core mimics the kinase inhibitors exactly to specific kinase inhibitors in its binding conformation. So what's the goal is the design and selective compound with selective affinity, selective PK profile, like the affinity against the cells. So moving on to next slide. So right, it's a little bit of background, biology background about what is kinase signaling pathway. I took an example here as a JAK1 inhibitor, just a general example I'm telling, as a JAK1. So JAK is Janus protein kinase, protein tyrosine kinase is an enzyme. So we'll have number of enzymes, these enzymes, JAK1, JAK2, JAK3, all these enzymes, they mediates the cytokinin signaling pathways. So one example you can see the left side is cytokinin binds to the receptor here. You can see just a simple pictorial representation. Anyone can understand. Cytokinin binds to the receptor. So then receptor dimerization here that will activate JAK phosphorylation. So JAK mediated phosphorylation, JAK is an enzyme, so that will activate the phosphorylation. And STAT is a protein here. STAT binds the phosphorylated receptor, and JAK phosphorylates the STAT protein, and the STAT dimer forms, and this dimer which can travel through the nucleus, and that will binds to the DNA. So once the DNA and binds to the DNA and activates, what will happen? Gene transcription, which causes number of diseases. It could be cancer, it could be rheumatoid arthritis, it could be respiratory diseases, any other diseases. So DNA transcription of specific target genes is if it happens, so that will modify the DNA in the DNA basis. Now moving on to next slide. Right. So what are the tricyclic target compounds we made? We made 4 series of compounds here. Number of compounds we made almost 20 compounds in each, 20 target specific compounds in each series, and worked on milligram to gram scale. So we explored the SAR, structural activity relationship, using a special software. So these compounds, so moving on to next slide. So I took the first one actually, 3 piperidine series here with the tricyclic core, which we made about 8 steps. And from these 8 steps, the last intermediate, we made number of compounds. R is a, the different substituents on the piperidine ring. We made about library of 30 compounds on each series. So the chemistry, I won't go near the in depth here. So the started with the imidazolpyridine. So the protection with phenyl sulfonyl, and then nitration, and then SNAR reaction, and then reduction, cyclization, and then deprotection, and then deprotection of benzyl group. This is the key intermediate. From this key intermediate, we made number of compounds, and these compounds, many of the compounds showed very good PK profile in the cells. So very active and very selective compounds. And then four piperidine series here with the different substituents which involve same chemistry. You can see the R is the number of compounds. Here these are selective compounds. We made the number of compounds, but these are all only selective compounds I'm showing here. And many compounds showed very good, excellent potency and testing against any other in vivo testing as well. So then other compounds we made is C2-hydroxyethyl series, and we explore this area as well. And with R is a different substitutions here. Made selective compounds, 18 compounds in these for the testing. And these cyno analogs, the cyno substitute analogs are very, very active against the cells, which are very good. We're very quite happy about those cyno compounds, and shows very consistent selectivity and cellular potency as well. And moving on to next slide, and we explore C2 exploration as well. So what we did is here, we took this intermediate and cyclized with hydroxymethyl, hydroxyethyl, acyl, and dimethyl hydroxy compounds as well, which shows very good selectivity. And these are all potent compounds which we tested. So what these are all. So these one more series, tricyclic series as well, and made put another nitrogen here, makes the compounds are very, very selective as well, very selective compounds, and cyno analogs are very potent compounds, shown very good PK profile. So all these compounds are in, so some of the very selective compounds are these are in advanced studies at the client place. So they're doing different various studies. There's a preclinical studies they did, and some of the compound enter into the phase 1 as phase 1, phase 2 as well. Because a lot of confidentiality of this, the studies, I can't show all the data at the moment. So moving on to next slide. So what is the conclusion of my project? So we're quite happy what we made, and the client was quite happy what they were doing. Like these compounds very progressing in advanced studies, and these are very novel compound, very selective potential kinase inhibitors, basically targeting the respiratory disease, targeting lungs, basically to open up the airways. And that is our target. So successfully synthesized a number of compounds, and we have significantly improved permeability. This is very important because the drug has to go through the plasma membrane, and showed very subnanomolar activities and excellent cellular potencies and very good PK profile. So this was my project. So now moving on to next slide, just wanted to give some health and safety tips on COVID, because I'm sure number of you knows all these tips, and you're following these. But just as many of the people, like as a participant in the seminar, so when then they see these pictorial representations, they might remember quite easily, and reduce the spread of the coronavirus. So basically, the first tip in anyone can tell you is washing your hands frequently with the soap water. So particularly specifically 20 seconds, because as I mentioned before, the virus has 70% fat. So when you washing your hands with a soap, that will decrease the interaction between the soap and hands. If you do the 20 seconds, if you feel difficulty with the washing long time, like 20 seconds, just sing happy birthday song for 10 times. That will be 20 seconds. So that's one tip. uh, then once you touch the contaminated objects, avoid touching nose, mouth, and eyes, and all those. So this will easily spread into the airways. And the other tips, I'm sure you're following this, do not travel if you are sick. Just stay home quarantine, and do not share food or drinks and personal items, including mobile phones as well. Mobile phones are another hotspots for the coronavirus, as the research suggested. And moving on to next slide. See, so he's wearing mask now. So because once it happens to him, so he's wearing mask. So wearing mask is quite important, as any mask, like reusable or disposable masks, as the previous speakers Ravi Kumar mentioned. And once it is disposable, once you use it, if it is disposable, throw it. Otherwise, if it is usable, just wash it and clean it properly. And stay home, stay safe, and stay positive. And avoid parties, all these birthday parties, the avoid functions and all those, avoid shaking hands and all those. Minimize the gathering with the friends and family. So until this pandemic situation disappears. and your family is very important. So you need to look after them as well. So you need to make sure all you got, like, rather than going out every time for the shopping, just you need to make sure you got enough things for a long time, like household supplies and medicines and all those. If you are sick, seek medical advice, like go to doctor and seek medical advice. Get flu vaccination, because in the winter is coming. These 3 months, like until January, it is a key time to spread. It might spread or might not spread, but we need to be very careful. And the other things you need to do is stay. If you have mild symptoms, just stay home quarantine. So when you're sneezing and coughing, you need to be careful with, like, you might spreading to other people as well. Use a mask or another. Use mask and use tissues and clothes, and throw them once you use them. Once you use them, and if your symptoms are worse, like, go to doctor. And you will be very stressful in this pandemic situation, because you can't meet anyone, friends and family. You might be like both physical and emotional stress you will have. Like, the look after yourself, do some regular exercise, keep in best possible health, take some healthy diet, go for a nature walk, make your time to relax, and communicate with all your friends and family as well. Ensure they are safe enough. And finally, I would like to thank Dr. Ravi Shankar and all other organizers and all other honorable members who are and all the participants um, uh, in listening, for their patience in listening my talk. And thank you for organizing this webinar. And I would like to thank Professor Ravi Kumar as
Thank you,
well
sir.
for agreeing this, giving this talk in such a short notice. Thank you, everyone. If you have any questions, I'll try my best to answer those. Thank you very much.
Any
Thank you, everyone.
questions?
Thank you, sir. Thank you.
No problem, sir.
Okay, thank you very much, sir, for sharing your useful insights with practical examples.
Thank you.
And we came to know one thing, that our C.V. Raman sir, means M.V. Raman sir, studied in Government Degree College, Sattupalli, Khammam district.
Yes,
Right,
yeah,
sir?
yeah. Yes, yeah.
Yes, it gives our students a lot of hope and confidence. And thank
Thank
you
you.
very much, sir, for
Yeah,
your
yeah.
nice presentation.
Yeah, thank you.
Yes.
So any questions?
Sir, good morning, sir.
Sir,
I have
yeah,
small question.
yeah, sir.
I have a small question, sir.
Yeah, okay, go on, sir. Carry on.
Sir, as you said, it takes about 2 years to prepare the vaccine.
Mm-hmm.
But some of the countries are saying that by the end of December or early 2021, they are preparing a vaccine. Is it
Mm-hmm.
possible? Whether all the phases were completed, whether the vaccine safety is okay or not?
Yeah, so as you see, like the previous news, like the Oxford vaccine or the vaccine in Russia. So they are like treating the people, but they are showing some side effects. I'm sure you heard about these side effects. Like the one of the person died in UK as well,
Yes,
isn't
sir.
it? So, and in the vaccine in Russia vaccine, like for example, every 10 members, one is getting allergy, like side effects.
Start with.
Hello, can you hear me?
Yes, sir. Yes, sir. You
Sir,
can
someone
please
is
continue, sir.
talking.
Yeah. So so I don't know whether so because because of the side effects, the Oxford vaccine, they stopped giving to the patients. So they need to correct dosage, and whether need to whether is there any virus replication or anything change is happening in the vaccine, they need to make the corrections first. They'll come out. So that's the reason I said, like, it's taking long time. So with short time, with rush, it's not good, or always not good actually, because it shows the side effects. Now if it shows any side effects, so that will kill the people. Isn't that, that's more disaster to the world? So that's why is vaccine is getting late. So I'm not sure whether it will come by December, but it will definitely take some time to come. Like they started in the last March, isn't it? So it takes some time to make the safe vaccine or safe drug. Making drug is different from making vaccine, because making drug to come into the market, at least it will take about 7 to 10 years to come to the new drug come to the market, to safe drug. Otherwise it won't be safe if it goes through like clinical trials phase 1, 2, 3, 4. It will be very difficult to approve the drug. And making vaccine, you can see like how the people how the companies are making vaccine, as I mentioned in my slide. It is easy to it can be it can be you can make the vaccine within the short time, but making vaccine is not a problem. But the thing is safety and efficacy, that's important. Like those are the challenging phases, like phase 1, 2, 3, preclinical phase 1, 2, 3, 4. So we don't know yet exactly is it 100% safe to put into the market, because that's the reason, because we didn't buy the Russia vaccine yet, isn't it? Because they said the vaccine is quite safe. So otherwise we could have bought for the millions of people if it is working really well. So and Oxford vaccine and AstraZeneca vaccine, they're still in clinical trials. They're still observing the people, as I mentioned, like the few hundreds people, few thousands people, still in Imperial College London and some other universities. They're doing some all these studies on the human beings at the moment. So once they are success, FDA can approve, then then they'll come into the market. Hope this answers your question. Hello.
Yes, sir. He's saying thank you, sir.
Okay.
Yes, he's satisfied with your answer. Okay, thank you,
Are you happy with my answer?
He's happy.
Hello. Hello.
Hello.
Can you hear me, Raman?
Yes, Ravi. Yeah. Yes, Ravi Kumar. Yeah.
It was a very nice presentation, although I am away from the organic chemistry nowadays.
Thank you.
What you have presented.
Mm-hmm.
So I have a question about, you mentioned several drugs in your presentation for the using coronavirus as antiviral.
Mm-hmm.
So, for example, several companies are developing various kinds of vaccines from their own slot.
Yeah.
For checking their own virus, I mean their own vaccine, they need several people according to your slide.
Yeah.
Am I right?
Yeah.
So, is the government will allow to check like that?
Oh yes, yeah. Because NHS always will have lots of data, and they will invite the volunteers. First, what they will do is they need to fill a lot of questionnaire, and they need to agree. Volunteers will need to agree to participate the studies. So number of people here, like they get volunteers. These are the people, especially in Western countries. They're all motivated about these actually, because when the NHS, all the people need to register at NHS here. So I'm not sure about the Korean. I'm sure about the same thing with Korea as well, with the GP. So they will have all the data about the patients. So health conditions and the backgrounds and community backgrounds and everything. So what they will do is this: they'll send the questionnaire to the people. Whoever is happy, they will participate in the clinical trials. So if now in Imperial College London, the hundreds of people are the volunteers are participating as volunteers. So I'm sure in the Western they have lots of people, because I may not know, like I may not go and see there, but they have special centers. What's happening there? Like they're making some clinical trials, AstraZeneca and the University of Oxford, and that doing all these trials. So yeah, number of people available. Yeah, definitely. Yeah.
So one person is only for one vaccine. Am I right? He cannot be duplicate.
Sorry, one person.
For example, I am the person can take only one vaccination as a voluntary.
Yes, yeah.
I cannot take other companies' vaccine as
No,
voluntary.
no, because they will select the people. They have to monitor every time what's happening. Is there any side effects? Is there any antibodies developed in the blood? Is your immune system developed? So they need to monitor that patient.
Okay.
Yeah.
Thank you for your kind answer.
Thank you. Good questions.
Sir, someone is asking about the second wave of corona.
No, that's not in my hands.
Okay, thank you.
Okay, no problem. Yeah, second wave. What's happening here is some of the areas which are highly infected, they are having second wave north of the UK. So the gradually spreading some areas. So there might be a possibility actually. So, but we don't know because that's the reason they completely locked down the north area, like Manchester and Wales and Northern Ireland. All those they are locked down at the moment. So they're quite highly restricted areas. You can't travel and you can't come out as well. And so there might be a chance actually this winter, but so that's why we need to be very careful in the winter season, especially. Yeah.
Thank you, sir. Thank you for your
You're welcome.
interactive session also. Okay, finally, I request Mr. M. Venkateshwar Rao, Assistant Professor of Zoology, to propose vote of thanks.
Thank you, madam. Thank you very much. Can I proceed?
Already over. Yes, sir. So continue.
Yes, madam. Honorable Sri Naveen Mittal IAS.
Sir, unmute yourself.
Can you hear me, sir?
Yes.
Thank you. Honorable Sri Naveen Mittal IAS, Commissioner, Collegiate and Technical Education and Chief Patron of today's webinar. Respected dignitaries, our most valued speakers, participating guests, it is my privilege to propose vote of thanks on this monumental event. I am M. Venkateshwar Rao, Assistant Professor of Zoology, on behalf of the college and entire team of organizers that have made this event a great success. On my own behalf, extend my vote of thanks to all the speakers for sharing their views and knowledge on the topic COVID-19, a pandemic of coronavirus, progress in therapeutic and vaccine development. It is a great honor that all the four internationally renowned speakers have very charitably agreed to render their talks in the webinar on the most relevant topic of the day. Before I go further, I must make a special mention of Sri Naveen Mittal, IAS, Commissioner, Collegiate and Technical Education. We at SR and BGNR Arts and Science Government Degree College, Autonomous, are greatly indebted to Sri Naveen Mittal for encouraging us, for helping us to organize these events like this. Thank you very much, sir. I express my gratitude to Sri Dr. Balabhaskar, AGO sir. We are thankful to you for your encouragement and gracious presence. I also extend our sincere thanks to Dr. J. Neerja Medangaru for extending her support all the academic events, activities. Sri Dr. Mahendra Diwal, Speaker of the Day, taking the opportunity to express my gratitude to each of you. I first thank Sri Dr. Mahendra Diwal for articulating his views on the intended topic for webinar. Dear sir, I thank you from the bottom of my heart for throwing light on the development taking place in the pharmaceutical industry to find a cure or vaccine for the most dreaded and worrying COVID-19 virus. As you said, Indian scientists could be the first ones to gift the world for the first vaccine, as most of them are in the third phase of testing vaccines, and they have been developed. I hope the great legendary of Usmanian it is on the way. Usmanian is on the way to prepare the synthetic antibodies against the COVID-19. Thank you very, very much, sir, for agreeing us. Dear sir, I don't know how to know my express my gratitude to you for sharing with us such a valuable information as now how through what the changes the COVID-19 virus evolved into its present fatal shape and how it is related to SARS-CoV-2 class of viruses. And Sri Dr. Veerabrahmachari sir, Head Department of Biotechnology, Krishna University, as you are have already observed, the strands in the COVID-19 virus are very similar to those in the SARS-CoV-2 also. And no wonder, COVID-19 could be a case of cloning that took birth in the laboratory and definitely not born from the animal food. Thank you very much, sir, once again. Dr. Ravikumar Chidrala, Research Professor, University of Ulsan, South Korea. Remdesivir is the only one hope for COVID-19 patients to sustain the attack. As of now, it is the only one drug approved by FDA for treating the COVID-19 patients, but scientists across the world are skeptical about the effectiveness in the saving the patients from the death. Dr. Ravikumar Chidrala sir talk how viable a treatment remdesivir could be in the COVID-19 case has cleared the doubts. And sir, thank you very much, sir. I sincerely thank you very much. Dr. N.B. Raman, scientist in drug discovery, Europhoenix Limited, United Kingdom. Our special thanks to Dr. N.B. Raman, sir, for sharing his views on development of inhaled drugs, target drugs, and the potential treatment for COVID-19. Your talk has been a very informative and useful. Thank you very much, sir. Our C.V. Raman of Satpalli went to United Kingdom. What a great journey, sir. Thank you very much for accepting our invitation. Thank you very much, sir.
Thank you.
Sri KSS Ratna Prasad, sir, SRN BGNR Degree College, Khammam. I now extend our sincere thanks to Sri KSS Ratna Prasad, sir, Principal, SRN BGNR Government Degree College, Autonomous Khammam, Telangana, India. Dear sir, you have been so kind and encouraging that I have no words to express my gratitude to you, but for your charitable and magnanimous permission for this program would have become such a big success. Thank you very much, sir. Thank you very much, and for supporting us. Srimati Anjani Saraswati Madam, IQSC Coordinator. I especially thank Srimati Anjani Saraswati Madam Garu, IQSC Coordinator. Thank you very much, Madam, for the invaluable support you extend throughout the webinar. I should say one word regarding pertaining to Madam. Behind every man's success, there is a woman, said someone. But I would say it is not only behind every man's success, it is the success of every nature. And thank you very much, madam, for accepting our invitation. And I go to Sri Tiruvengalachari sir, Principal Parkala, Government Degree College. And I also extend my sincere thanks to Sri Tiruvengalachari sir, Principal Parkala Degree College. Dear sir, your guidance has been a great help to us in conducting this webinar successfully, and with all due respect, I thank you very much, sir. I sincerely thank Sri Ramesh sir, Assistant Professor of Department of Commerce. I took the responsibility of ensuring the best and uninterrupted webcasting of the event. Dear sir, please accept my thanks. I express our gratitude to Sir Dr. P. Ramesh Garu, Head Department of Chemistry, and thank you very much, sir. I also thank Dr. Chaitanya Madam, Assistant Professor of Botany, for her support. I thank you very much, Madam. I express our special thanks to Srimati P. Jyoti Madam Garu. Well beginning is half done. A well beginning is half done. You really gave this webinar a right beginning with a very good song with your sweet voice, and that ensures success. I also extend my sincere thanks to Dr. Samaya sir for his beloveds in letting us make use of their study center for this webinar. I also extend my lot gratitude to Sri Katta Srinivas Garu for his technical support along this day. Thank you very much, sir. Thank you very much, sir. Last but not the least, I extend my special thanks to Dr. C.H. Srinivasulu, sir, convener of the program, convener of this event, and Sri Dr. V. Ravishankar, sir, assistant professor, Department of Zoology, and coordinator of this webinar, and my colleagues and my other colleagues of Department of Zoology. I sincerely thank each and every member and each and every faculty member of the legendary college in the Telangana state, SR and BGNR Government Degree College. I thank you very much, sir. And I also say one thing, sir. With these words, I announce that this webinar has come to an end. The world is passing through one of the most terrible periods of a crisis in the history. I wish everyone to be healthy, happy, and safe living. Stay home and stay safe. Set example to others being safe. Thank you very much.
I request all the participants to fill the feedback form given in the chat box. So you'll get the certificates as soon as possible. Now let's call it a day. Bye.
Bye-bye. Thank you.
Bye.
Thank you, sir.
Thank you. Well done. Well done, everyone.
Thank you very much, sir. Thank you very much, sir.